Hey champs of Examsnet, welcome back to the series of chapter-wise discussion. We are back with another chapter, that is biomolecules. In the previous session, we have completed the chapter Cell, the unit of life, right? Cell, the unit of life. Today, we will be completing this chapter, Biomolecules. And in the next session, we will be completing the chapter Cell Cycle and Cell Division. Cell cycle and cell division. Now these three chapters are actually part of the same unit, okay? These are one unit because all these three chapters are related to each other, okay? How this chapter biomolecules is related to cell? We have studied that cell is the fundamental unit of life, okay? It is the structural and functional unit of life. So we know that the cell is the building block of living organisms. Building block of living organisms. Similarly, the biomolecules are the building blocks of a cell, okay? Biomolecules are building block of a cell, okay? Bio means life and molecules are small unit of chemical compounds, right? Molecules are small unit of chemical compound. What are the examples of biomolecules? The examples are proteins, carbohydrates, lipids, nucleic acids, etc. Okay. So we know that the cell consists of these biomolecules. We have seen that the cell membrane or the plasma membrane was made up of lipid and protein, right? It was made up of lipid and protein. We have discussed about the integral protein and peripheral proteins, right? Similarly, we have seen that the ribosome, which is a non-membrane bound organelle of the cell, is involved in the synthesis of protein, okay? Protein synthesis. So can you see that these lipids, proteins, these are part of the cell, right? These are important parts of the cell. Without them, the cell will not be functional. Then another important thing, nucleus. We know that the nucleus consists of the genetic material, that is DNA. So what is DNA? DNA is a nucleic acid, which is again a biomolecule, right? So these are biomolecules which are involved in all the functions of a cell. Without biomolecules, the cell will be useless because it will not be able to function, right? Therefore, biomolecule is totally related to the chapter cell and it comes under the same unit. Okay, so let's start this chapter. So the first topic under this chapter is elemental analysis. That means analysis of all the elements which are present in a living tissue. Okay. So after performing many experiments, it was concluded that all the elements which are present in a sample of Earth's crust are also present in a sample of living tissue. That means the overall composition of a living tissue is same as the composition of the Earth's crust. Okay. But there will be difference in their percentage composition. Okay, so the amount of these elements will vary. Okay, the amount of the elements will be different in living tissue and in the non-living, that is the Earth's crust. Okay, but they will be present definitely in both of them. So here we have a comparison of different elements present in the Earth's crust and in the living tissue. Okay, so here we will have a comparison of the percentage weight of the elements present in these two, living and non-living. So for hydrogen, the first one, in the Earth's crust you can see what percentage of hydrogen is present. 0.14% hydrogen is present, whereas in case of human body the percentage composition of hydrogen is 0.5. so in which case it is more? It is more in case of human body. Okay, So in case of human, the percentage of hydrogen is more. Then the next one is carbon. Okay, so you can see that the percentage composition of carbon in the non-living, that is the Earth's crust, is only 0.03, whereas in case of living, that is human body, is 18.5%. That's a lot. Okay, so again, the percentage of carbon is also more in case of human body than the Earth's crust. Next is oxygen. Here you can see in the amount of oxygen or the percentage of oxygen in the Earth's crust is also large. Okay, it is 46.6%, but it is slightly more in case of human body, that is 65%. So again, the amount of oxygen is also more in case of human body. Nitrogen is very little in the Earth's crust and it is about 3.3% in the human body. Again, nitrogen is also more in the human body. Next is sulfur, 0.03 in case of Earth's crust and 0.3 in case of human body. Which one is more? 0.3 is more. So again, the sulfur is also present in larger amount in case of human body than the Earth's crust. Okay. So these things you can see: hydrogen, oxygen, carbon, nitrogen, sulfur. Okay, hydrogen, carbon, oxygen, nitrogen, sulfur, these are the things which are present in larger amount in case of human body, that is in the living body, than the non-living, that is the Earth's crust. Okay, whereas there will be some elements which will be available in larger amount in the Earth's crust, like you can see sodium. What is the amount of sodium in human body? Only 0.2, whereas in case of the Earth's crust it is 2.8. You can see this is larger amount. 2.8% is more than 0.2. So sodium, then we have calcium, magnesium, silicon, all these things are present in larger amount in Earth's crust and they are present in very less amount in the human body. Okay, so some elements are present in larger amount in Earth's crust, some other are present in larger amount in the human body, but all these are present in both living as well as non-living. The only difference is in percentage composition. Somewhere they can be more in amount, somewhere they can be less in amount. Okay, so this was about the elemental analysis. And here you can note that oxygen is the most abundant. Okay, what is the maximum percentage of elements that is present in the human body? 65%. You can see that is oxygen. So what is the most abundant element in the human body? Oxygen. Okay, oxygen is present in water also, right? We know that about 70% of our weight is by water. So water consists of oxygen, right? Then we come to the chemical analysis. So what is chemical analysis? Here we will analyze the chemicals, the amount of different chemicals which are present in our body. So for this also an experiment can be performed. Now in this experiment a tissue sample is taken. Okay, it can be any living tissue sample like liver or vegetable. Okay, then this tissue sample has to be grinded. Okay, we have to grind this tissue so that we can break the tissue into very small pieces so that the substances, the chemical substances can be released from the tissue. Okay. So this will be grinded and a liquid will be used, that is trichloroacetic acid. Okay, this is similar to the grinding of spices. At home also we grind the spices in kitchen, right? So what do we do when we have to prepare a powder? Then we don't add any liquid to it. But when we have to make a paste or a thick slurry of the spices, then we have to add some liquid. Okay, so here also we have to prepare a thick slurry. Therefore, we will have to add some liquid. So the most suitable liquid for this is trichloroacetic acid. So you have to remember the name of this acid that is used in this experiment. Okay. So a thick slurry is prepared. Now what is the use of this thick slurry? Here the acid that we have used, trichloroacetic acid, will be able to divide the chemical substances into two fractions. One will be the acid soluble fraction and the other will be acid insoluble fraction. Okay, soluble means they will be dissolved in the acid, they will be soluble in the acid, and then there will be some chemical substances which will not be dissolved in the acid, so they will come under the acid insoluble fraction. Okay, so we have to separate these chemicals now. These chemicals have been released from the tissue after grinding. Now they have to be separated into two parts. So for that we use cheesecloth or cotton cloth. Okay, any one cloth can be used, cheesecloth or cotton cloth. What will be the role of this cloth? It will act as the filter medium. It will act as the filter medium that will help us the filter out some substances from the other. Okay, so when we use, suppose this red color is the cheesecloth or the cotton cloth. So the thick slurry will be made to pass through this cloth. What will happen? Some of the substances present in the slurry will be able to pass through this cloth. Okay, because we know that the cheesecloth or the cotton cloth will have very minute pores in it. Okay, so these small pores will allow the passage of some chemical substances, and they will block the passage of some other substances. Okay, so which some substances will be able to pass through it, which will be very small in size. Okay, so very small sized substances will pass through this cloth, and they will be the filtrate. These are known as acid soluble pool because these were dissolved in the acid, therefore they have passed through the cloth. Okay, so these are known as acid soluble pool. These are also known as filtrate because these are filtered out from the cloth. Okay, and since these are very small in size, that is why they are able to pass through the cloth. These are also known as biomicromolecules. Okay, micro means very small molecules. Bio is added because here the tissue is living tissue, right? So bio means living, life or living. So here these are biomicromolecules, very small sized biomolecules, and it will also contain some inorganic ions or some salts like sulfate, phosphate, etc. Okay, so these will be part of the acid soluble pool. Then there will be some other substances which will be larger in size, therefore they will not be able to pass through the very minute pores of the cloth. So they will remain on this cloth. These yellow colored particles you can see, these could not pass through the small pores of the cloth because these are larger in size, or we can say these are macromolecules. Okay, so these are biomacromolecules. Biomacromolecules, and these were insoluble in the acid, therefore these could not pass along with the acid. Okay, so these come under the acid insoluble pool. So did you understand? When we made a thick slurry of the tissue, then we passed this slurry through the cheesecloth or cotton cloth. Then some substances which were dissolved in the acids, they could pass through the cheesecloth and they come under the acid soluble pool. Then there will be some other substances which are larger in size and they were insoluble in the acid, therefore these could not pass through the cheesecloth. So these are acid insoluble pool, and these are also known as retentate because these were retained on the cheesecloth, they could not pass through this. Okay, so the acid insoluble pool, which are also known as retentate, which could not pass through the cloth, suppose this is the cheesecloth, so these substances could not pass through the cloth because these were larger in size, or we can say these were biomacromolecules. Okay, so these biomacromolecules can be proteins, these can be polysaccharides, these can be nucleic acids, or they can be lipids also. Okay, so all these different types of biomacromolecules are retained on the cheesecloth or the cotton cloth. Now when we have to study about each one of them, suppose we have to study about protein or we have to study about the polysaccharide, then we will have to separate this biomacromolecule from the rest of the biomacromolecules. So how can we separate them? By using different separation or purification techniques. Okay, so different types of separation or purification techniques are available by which we can separate these, and then we can study the biomacromolecules which we want to study about. Okay, so this was about the chemical analysis. Okay, and these are the biomacromolecules which we will be studying in this chapter. Okay, there is another type of analysis known as ASH analysis, which is used for the analysis of the inorganic materials, inorganic substances or inorganic materials. Okay. So the chemical analysis was about the analysis of the chemical substances or the organic substances, okay, because the lipids, proteins, polysaccharides, all these consist of the organic molecules, right? Whereas we know that in the acid soluble pool, which was able to pass through the cloth, that used to contain the inorganic substances. So if you have to study about those inorganic substances present in the acid soluble pool, then you can use the ash analysis. Okay. Now here what we have to do, we have to take our tissue sample which contains water. Okay, so initially it will contain water. So this is the wet weight of the tissue. Then we will have to dry it to remove the water present in it. Okay, so when you dry it, the water will be removed and then you will get the dry weight. Okay, now this dry tissue has to be burnt. So what happens when you burn this tissue? When you burn this tissue in the presence of oxygen, what happens when you burn something? Oxygen is supplied to it. Okay, so this oxygen that is supplied to it will connect with the carbon present in it, and this will become carbon dioxide. It will be removed in the form of gas. Then this oxygen can combine with the hydrogen present in the sample, and then it will make water, H2O. Then this water will also be released from the tissue. Okay, so this way all the organic molecules, the elements which make the organic compound, which these will be eliminated. Okay, so what will be left eventually? Finally, only the inorganic substances will be left. Okay, so the ash that is formed after burning will consist of only inorganic ions. Okay, because we know that the organic compound mainly consists of carbon, hydrogen and oxygen, right? Methane, ethane, all these things. So carbon will be combined with oxygen, it will form carbon dioxide, it will be removed from it, okay, in the form of carbon dioxide gas. Hydrogen present in the sample will again combine with the oxygen, it will form water, it will also be removed. Okay, so only thing that will be left in the ash will be the inorganic ions. Okay, so if you have to study about the quantity of or the amount of inorganic ions present in a sample, then which analysis you have to use? You have to use ash analysis. And when you have to study about the living the organic molecules, then you will have to use the chemical analysis. Okay, now when we saw the chemical analysis, here you can see we have a chart. This table, you can see this table was given in the NCERT, so I've added this. Here we have some components like water, proteins, carbohydrates, lipids, nucleic acids, and the percentage of the cellular mass, because these are present in the cells. Okay, these are the substances which are found in the cells. So in what percentage or in what amount they are present? Water is present in about 70 to 90%. Okay, proteins 10 to 15%. Carbohydrates 3%, lipids 2%, and nucleic acids 5 to 7%. Okay, so this is the percentage composition of the cell. Now the next. Okay, so we saw the ash analysis, right? What is ash analysis? It was used for the to find out the amount of inorganic ions present in a living tissue. So these ions can be sodium ion, potassium ion, calcium ion, magnesium ion. Then water has been removed from it. Okay, this water is removed. Then some salts can also be present like sodium chloride, calcium carbonate, phosphate, sulfate. So all these things will be found in the acid soluble pool. Okay, these are found in the acid soluble pool. Always remember this. Okay, so these were filtrate because they were able to pass through the cheesecloth or cotton cloth. Okay, whereas all the macromolecules like proteins, polysaccharides, then nucleic acids, all these things were found in the acid insoluble pool because of their large size. Okay. So we can say that the biomolecules are classified into two groups. One is macromolecules and the other is micromolecules. Okay, the word macro means large and the word micro means small. Okay, so obviously the macromolecules will be larger in size. These will be present in the acid insoluble pool, and the micromolecules are smaller in size and they were present in the acid soluble pool, acid soluble pool, and they were able to pass through the cloth. So what is the weight of macromolecules? It is generally more than 1000 Dalton. Okay, so the weight of the biomacromolecules will be larger than 1000 Dalton, whereas the weight of the micromolecules will be less than 1000 Dalton, and generally it is between 18 to 800 Dalton. What are the examples of macromolecules? Here we have proteins, polysaccharides, nucleic acids, and lipids. And the examples of micromolecules are amino acids, nucleotides, which are the monomers. Okay, amino acids, they combine with each other and they form the protein. Okay, so amino acid, when undergo polymerization, they will form the protein. Nucleotides, after polymerization, will form the nucleic acids. Okay, nitrogen bases are also present in the nucleotides. Sugars and inorganic ions, all these are parts of the acid soluble pool, and these are macromolecules. Now one thing that you have to note is lipids. Here you can see lipids is present among the macromolecules. Okay, but do you know that the weight of the lipid is similar to the weight of the micromolecules? It is not more than 1000. Okay, the weight of the lipid is less than 1000 Dalton. Less than 1000 Dalton. Then why is it present among the macromolecules and not among the micromolecules? The reason is that it is generally found along with the macromolecules. Okay, so although its weight is less, but it was found in the acid insoluble pool. It was not found in the acid soluble pool. Okay, so there are two methods. One is weight, but one you can classify them on the basis of weight. Second, on the basis of their solubility in the acid in the given experiment. Okay, so all the substances or all the molecules which were present among the acid insoluble pool were classified under biomacromolecules. So lipids, although it has lesser weight, around similar to the micromolecules, but it was found among the macromolecules in the acid insoluble pool. Okay, because we know that it is part of the cell membrane. Okay, we know that the cell membrane consists of proteins, lipids, and carbohydrates, right? So when we are grinding a tissue, that means when we are disrupting the cellular structure. Okay, so the cell membrane will also be disrupted. So all the compositions, all the components of the cell membrane, like proteins, polysaccharides, and lipids, all these will be collected at the same place. Okay, so therefore the lipids were also found along with the proteins and polysaccharides. So this is the reason why they are placed under macromolecules. Okay, now here you have to remember that suppose the question asked strictly according to the weight, then if the question is according to the weight only, then lipid will be placed under the micromolecules. But if in general it is asked, then it will be placed under the macromolecules. Okay, or if it is specifically asked about its solubility in the acid, trichloroacetic acid, then also it will be placed among the macromolecules, the larger molecules. Okay, now all these amino acids, nucleotides, nitrogen bases, all these things are the cytoplasmic composition. Okay, cytoplasmic. These are found in the cytoplasm of the cell. Whereas the macromolecules like proteins, polysaccharides, lipids, all these are the structural component of the cell. These are structural component. We know that the cell membrane consists of protein, polysaccharides, lipids. Nucleic acid is present in the nucleus, okay, in the genetic material DNA. Okay, we know that DNA is what? DNA is nucleic acids, and DNA is present in the nucleus of the cell. Okay, so together these form the cell composition. So all these things can be found in the cell. So I hope things are clear till now. Now we come to different types of metabolites. Okay, metabolites, what are metabolites? These take part in the metabolic reactions. these take part in the metabolic reactions. Okay, what is metabolism? Metabolism is the sum total of all the chemical reactions, going on inside our body. Okay, so these chemical reactions can be catabolic reaction, or they can be anabolic reaction, right? So all the metabolites take part whether in the catabolic reactions or the anabolic reactions. Okay, that means they take part in the metabolism of the body. So these metabolites can be of two types. First, primary metabolite and the secondary metabolite. Okay, now what is the difference between the primary metabolite and the secondary metabolite? Primary metabolites are those which will further take part in the metabolism. Okay, so they will further participate in the metabolism, and they have identifiable role in the body. Next is they help in normal physiological functions of the body like growth, development, reproduction, or in normal survival of the body. Okay, so they will have an important role in the physiological functions of the body. Examples of the primary metabolites are glucose. We know that the glucose provides energy to the body, right? Amino acid polymerized to form protein. We know that protein has a very important role in our body. Okay, then what are secondary metabolites? Secondary metabolites are those which have exit the metabolic pathway. Okay, they have no further role in metabolism of the body. Okay, so no participation in the metabolism anymore. Initially they might have participated, but now after they are finally created, now they will no more participate in any metabolic reaction. Okay, so they have no identifiable role in the body. So you have this chart which shows you the examples of the secondary metabolites. So these secondary metabolites can be present in the form of pigments like carotenoids, anthocyanins, etc. Okay, so although they give some color, to the plant or fruit or vegetable, but they do not participate in the metabolism. Okay, they have no identifiable role in the body. Okay, then they can be present in the form of alkaloids like morphine, codeine, etc. They can be terpenoids like monoterpenes, diterpenes, etc. Then they can be present in the form of essential oils like lemongrass oil, etc. So see, these things can be beneficial for us. Okay, they can be beneficial in ecology, but they will not be of any benefit to the plant body in which they are formed. Okay, then they can be present as toxins like aberrin, ricin. They can be present as lectins like concavalin A. Then they can be present in the form of drugs like vinblastine drug, curcumin drug, etc. Or they can also be present as polymeric substances like rubber, gum, cellulose, etc. So you will find many uses of these secondary metabolites. For example, cellulose. Cellulose is present in the plant body. Okay, we know that the cell wall consists of the cellulose. Okay, so it has no role in the metabolic reactions that are taking part in the plant's body. Okay, rubber, we can use rubber for many purposes. We extract the rubber from the plant, then we can use them in many forms. But does this rubber take part in the metabolic reactions? No. Okay, it has no role, no identifiable role in the plant in which they are formed. Okay, similarly gums also, all these things can be useful for us but not for the plant in which they are formed. Okay, so sometimes the direct questions are asked from this table. Okay, if the question is asked from this topic, primary and secondary metabolites, then most of the time the question is from this table only. So you have to remember the names and to which category they belong. Okay, you have to remember that morphine and codeine belong to the alkaloids. You have to remember that concavalin A is a lectin. Okay, so all these things you have to remember. This table is very important for you. Okay, now we will move to the next topic. Now amino acids. Amino acids, it means they will have one amino group and one acidic group, that is carboxyl group. Okay, so NH2 group and carboxyl group COOH, these two will be definitely present in the amino acids. Now these amino acids are nothing but the substituted methane. What is the structure of methane? We have one carbon, then 4 hydrogens attached to this carbon, right? This is the structure of methane. So amino acids are substituted methanes. That means the hydrogens of the methane are substituted or replaced by some other groups. So one hydrogen will be replaced by the amino group. Another hydrogen will be replaced by the carboxyl group COOH, and the third hydrogen will be removed by some R functional group. Okay. Now the amino group and the carboxyl group, these two groups will be constant in all the amino acids, but the R functional group will be variable. Okay, this R group is variable. This will vary from one amino acid to next. Okay, so the nature of functional group R can be different in different amino acids, right? So here you can see one hydrogen has been replaced by amino group, another hydrogen replaced by the carboxyl group, and this third hydrogen is replaced by the R functional group, which is the variable group. Okay, so now can you say that the amino acid is nothing but the substituted methane? Okay, now these are also known as alpha amino acids because the amino group and the carboxyl group, these are attached to the alpha carbon. Okay, amino group NH2. Okay, NH3 plus is formed after gaining a proton. So amino group and carboxyl groups are attached to the alpha carbon. What is alpha carbon? The carbon to which the functional group is attached. Okay, so here we have only one carbon to which amino group is attached, a carboxyl group is attached, and the R functional group is also attached to the same carbon. Okay, so this is the alpha carbon. Therefore, the amino acids are also known as alpha amino acids. Okay. Then the chemical and the physical properties of amino acids are essentially of the amino group, carboxyl group and R functional group. That means the nature or the properties of the amino acids will directly depend on the amino group, carboxyl group and the functional group. Okay. So depending on the amount of or number of acidic groups and basic groups, the amino acids can be either acidic in nature or they can be basic in nature. Okay, if the number of acidic group and basic groups are same, or if the overall charge on the molecule is 0, then the amino acid will be neutral. Okay, here we have the examples. What are the examples of acidic amino acid? Glutamic acid. Then aspartic acid can also be the example. Aspartic acid. Then we can have basic amino acids, for example lysine. We can have arginine, histidine. All these are basic amino acids. Then we can have neutral amino acids like valine. Then we can have alanine. Alanine is the amino acid in which the R functional group is methyl group. Okay. Then we can have glycine. This is the simplest amino acid. Glycine is the simplest amino acid. So we have seen that the R functional group varies, right? So if this R functional group is simply a hydrogen, then the amino acid is known as glycine, which is the simplest amino acid. Okay, this is the simplest amino acid. If this R functional group is methyl group, CH3, then the amino acid is alanine. Both these glycine and alanine are neutral amino acids. Okay. If this R functional group is CH2OH, then what will be the amino acid? It is known as serine. So as the functional group is changing, the amino acid is also changing. Okay. So different amino acids will have different R functional group. I hope you understood this. Then we have some examples of aromatic amino acids like tyrosine, phenylalanine, tryptophan, etc. Okay, they will contain some aromatic groups. Then we can have sulfur containing amino acids also. So it is your task to search about these amino acids, okay, and see their structures. So when you will see their structure, then you will understand them, these things better. Okay, why they are known as acidic amino acids, why these are basic, why they are called aromatic amino acids. For example, if you search the structure of tryptophan or phenylalanine, there you will see that the aromatic groups are attached to them. Okay, so you will understand and you will remember that these are aromatic amino acids. Okay. Now, one of the very important point is the ionizable nature of the amino and the carboxyl group. So these two groups are ionizable in nature. That means they can be ionized. That is, they can acquire positive or negative charge. Acquire positive or negative charge by the exchange of electrons or protons. For example, here you can see this is the first structure. 3 structures are shown here. This is the first structure. Now what is the condition here? We have low pH. So under a low pH condition, we have acidic condition, right? Acidic condition. We know that pH is equal to minus log of H plus ion concentration, right? Minus log H plus ion concentration. It means as the pH will decrease, the concentration of H plus ion will increase. Okay, so we are talking about the acidic condition. That means low pH condition. Low pH condition means higher concentration of H plus ions, right? So when the concentration of H plus ion, that is proton, is higher in the medium, then what will happen? The amino acid will acquire the proton, okay, because the concentration of a proton is higher, so some proton will get added to this molecule. So we have 2 locations, right? We have carboxyl group and amino group. Amino group is basic in nature, okay. This also you have to remember that the amino group is basic in nature. And we know that the bases accept proton more readily. Okay, they accept proton, H plus ion. And the carboxyl group is acidic in nature. And we know that the acids donate proton more readily. Okay, so they donate proton or they release proton. So in the first case, when the pH is low and the concentration of H plus ion is higher, so the proton has to be accepted. So who will accept the proton? Amino group or the carboxyl group? Amino group will accept the proton because this is acidic in nature, this is basic in nature, and we know that the bases are proton acceptors. Okay, so the amino group, that is NH2, will accept a proton and it will become NH3 plus. Okay. Initially 2 hydrogens were there, now 1 more hydrogen, so H plus. Okay, so NH3 plus. So what is the overall charge on the molecule in the first case? Positive, because there is no negative charge here. Okay, so the overall charge on the molecule is positive. In the second case, you can see we have neutral condition. Now if we compare the neutral condition with the acidic condition, here the pH will be slightly higher. Okay, so here the pH will increase from the earlier condition. Okay, so as the pH will increase, the amount of H plus ion will decrease. Okay, H plus ion concentration will decrease. So now the proton has to be removed from the molecule. So you can see that the proton is connected with the carboxyl group as well as the amino group. So who will lose the proton first, or who will donate the electron first? So we know that the acids are the proton donors. Okay, so the acidic group, that is the carboxyl group, will donate the electron first. So COOH will remove the electron and it will become, it will remove the proton and it will become COO minus. Okay, and NH3 plus will remain as it is. So now you can see that we have 1 positive charge and 1 negative charge. So the overall charge on the molecule will be 0. So the molecule will be neutral in nature, and this molecule is known as Zwitter ion. Okay, this is known as zwitterion. It means that the amount of positive and negative charge is equal, so they will cancel each other out, so the overall nature will be neutral. Okay, then in the next case we have higher pH, that is basic condition. So what will happen in this condition? In basic condition, the pH will further increase, so the concentration of H plus ion will further decrease, right? So more H plus will need to be removed from this molecule. So carboxyl group has already removed the proton. Now there's the chance of amino group to remove the proton. So now the amino group has to remove the proton. It was NH3 plus, now it will remove the proton and it will become NH2. So there will be no charge on the NH2, but there is one negative charge on the carboxyl group. So the overall charge in this molecule is negative, right? So the overall charge is negative. Okay, so this was about the this was just the explanation of the ionizable nature of the amino and carboxyl group, how these get ionized. Okay. So you have understood about the amino acids, right? Now what will happen if a large number of amino acids are connected together? They will form protein. Okay, so protein is the next topic, and protein is formed by peptide bond formation between two adjacent amino acids. Okay, so suppose this is one amino acid. Okay, this is one amino acid. This is another amino acid. If a peptide bond is formed between these two, that means two amino acids are connected together by a bond, then it will form dipeptide. Dipeptide means two amino acids are connected together by the peptide bond. Okay, what is peptide bond? We will see later. First, we will have a discussion on general characteristics of the protein. So here I am just giving you an introduction about the peptide bond. Okay, so peptide bond is the bond that is formed between two amino acids. Now if only two amino acids are connected together, it is dipeptide. This structure will be known as dipeptide. Suppose one more amino acid will be connected to it. So how many amino acids now? 3 amino acids are there. That means the complete structure when 3 amino acids are connected together by peptide bonds will be known as tripeptide. Tripeptide. Okay, tripeptide means 3 amino acids are connected together by the peptide bond. Okay, similarly, if the number of amino acids increases further, it is 4 amino acids, 5 amino acids, 1000 amino acids, lakhs amino acids. Okay, so if the number goes on increasing, it will become polypeptide because a large number of amino acids will be connected together. Okay, then it will become polypeptide. And polypeptide is nothing but the protein. So another name of protein is polypeptide because it consists of a large number of amino acids connected together by the peptide bond. Okay. So protein is a heteropolymer. Heteropolymer means different monomer units are attached to it. Okay, that means the amino acids which are connected together to form the protein can be different. It is not compulsory that all the amino acids will be same. We have seen that different types of amino acids are there. For example, glycine. We can have alanine. We can have serine. Okay, so all these different types of amino acids can also be connected together to form a protein chain, right? Therefore, it is known as heteropolymer. Okay, it's not that only glycine will be present in a chain or only alanine will be present. Different amino acids can be present in a single chain. Therefore, it is known as heteropolymer, consisting of different amino acids. So, it is formed by different amino acids. Got it? What are the functions of the proteins? So there are numerous functions of the proteins. Okay, proteins can be present in various forms and they can perform variety of functions. For example, here are some of the functions which are mentioned, like transport nutrients across the cell membrane. So when they are present as membrane protein, okay, they can transport materials across the cell membrane. Then they can fight infectious organism when they are present in the form of antibodies. Okay, when they are present in the form of antibodies in the body, then they help in the immunity. Okay, so they fight infectious organism. Then they can also be present in the form of enzymes. So when they will be present in the form of enzymes, what will they do? They will catalyze the biochemical reactions going on in the body. Okay. So these are only some of the examples. There are variety of functions which can be performed by the proteins. Okay, and the proteins can be present in different forms depending on the situation or the requirement. Okay. Here you have to remember facts like which is the most abundant protein in the animal world. So this is collagen. This question is often asked, which is the most abundant protein in the animal world? Collagen. But this is the abundant protein only in the animal world, not in the plant world. Okay. So which is the most abundant protein in whole biosphere, including plants and animals? It will be rubisco. Rubisco, it is an enzyme. Okay, so this is the protein which is present in the form of an enzyme. The name is rubisco, and it is the most abundant protein in the whole biosphere. Okay, we will be studying about this enzyme in the chapter photosynthesis. Okay, we will be discussing this chapter within next 2 days. Okay, there we will understand it better. Okay. In plant physiology, we will be studying about the rubisco enzyme. Then here are some of the proteins and their functions, like collagen. What is the function of collagen? So it acts as the intercellular ground substance. Trypsin is the protein in the form of an enzyme. Okay, so trypsin is an enzyme. Then protein can also be present in the form of hormone, for example, insulin. So insulin is a hormone which is again a protein. It can also be present as an antibody, then it will be involved in the immune system of our body, and then it will fight the infectious agents. Then it can be present as receptor. So there what will it do? It will help in the sensory reception like smell, taste, hormone, etc. Okay. Means it will help the hormones. Hormone itself is a protein. Now to help the hormones there are receptors. Okay, receptors are available where the hormones can act. Okay, hormones act through the receptors. So the receptors are also a form of protein. Now GLUT4, GLU means glucose. GLU is a short form of glucose. T means transfer. So they help in transferring of glucose. So they enable the glucose transport into the cell. Then they are in the form of GLUT4. Okay. So you will be studying about each of these in different chapters. Here we just have to see the examples of different types of functions which can be performed by the protein. Next is peptide bond formation. We have seen that the bond that is formed between different amino acids to form the protein is the peptide bond. So how is a peptide bond formed? Here you can see we have two different amino acids. This is one amino acid. Okay, here we have NH2. This side we have COOH, carboxyl group. This is the second amino acid. Again we have this amino group here and carboxyl group here. Now you have to remember, you have to keep in mind that when the bond formation has to take place, then the reaction will take place between the amino group of one amino acid and the carboxyl group of another amino acid. Okay, so amino group and the carboxyl group. Okay, the reaction will take place between the amino group of one amino acid and the carboxyl group of another amino acid. That this reaction will not take place between two amino groups or two carboxyl group. Okay, one group must be amino group and the other group must be carboxyl group. Okay, so what will happen? Amino group means NH2. Okay, so NH2 and the carboxyl group is COOH. Now, if you look here, this is the carboxyl group. From the carboxyl group, this OH will be removed, and from the amino group, one hydrogen will be removed. So what will be left here? Only NH will be left, and this side CO will be left. Okay, so the peptide bond will be C double bond O, and this side NH. Okay. So this is the peptide bond. This is how the peptide bond will look. And as you can see that from the carboxyl group, OH has been removed. From the amino group, one hydrogen has been removed. So now we have H2O. So H2O, that is water, will be released from this molecule. Okay, that means dehydration is taking place. So this reaction is a dehydration reaction. Okay, the formation of polypeptide, okay, or the peptide bond formation is a dehydration reaction. That means it includes or involves the removal of water. Okay, I hope you have understood this. Now another important thing that you have to remember is that suppose this is the polypeptide chain. Okay, polypeptide chain, that means amino group, carboxyl group. Okay, so each amino acid will have the amino group as well as carboxyl group. So in this chain, at one end amino group will be free, and on the other end carboxyl group will be free. Okay, in a polypeptide chain, one amino acid will be free. Okay, one end will be free, and that will be containing the amino group. So this is known as N terminal. And the other end of the polypeptide chain, where carboxyl group, carboxylic acid, is free, there it is known as C terminal. Okay, so the polypeptide chain has one N terminal consisting of amino group and the other C terminal consisting of carboxyl group. Okay, I hope you have understood this reaction well. It is very important to understand this reaction. Here you can see the bond formation is shown in the blue color. Okay, so you have to focus in blue color. You can see that OH has been removed from carboxyl group. This side H has been removed from the NH2. So H2O has been removed from it. Okay, and then a bond is created between carbon and nitrogen, carbon of one amino acid and the nitrogen of another amino acid. Okay, so this bond is CONH. This is the peptide bond. Okay, and here you can see that the two amino acids between which the peptide bond is forming, here the amino acid is same. Okay, so R functional group is not mentioned. This R functional group can be different in both the amino acids. For example, in the first amino acid it can be H if it is glycine. In the second amino acid, if suppose this is alanine, so it can be CH3. Okay, so two different amino acids are there. Peptide bond is forming between two different amino acids. Therefore, it is known as heteropolymer, right? So I hope it is clear why the protein is known as heteropolymer. Now we will have a look at the structure of protein. Now we know that in chemistry we understand the structure in a two-dimensional forms. Okay, we study the two-dimensional structure of molecules. In physics we study the three-dimensional structures, whereas in biology, especially when we are studying about the proteins, we study the four-dimensional structures. Four-dimensional structure of the protein. Okay, so the protein can have different types of structures. It can have primary structure, it can have secondary structure, it can have tertiary as well as quaternary structure. Okay, so primary structure. How does a primary structure look? It will be simply a chain consisting of different amino acids which are connected by the peptide bond. Okay, so here you can see each of the blue boxes show one amino acid. Okay, so one box means one amino acid, another box another amino acid. So you can see different amino acids are connected by the peptide bond in the form of a long chain. So it will look something like this. The primary structure will be simply a chain of amino acids connected by the peptide bond. So this is polypeptide chain. This is the primary structure. Okay, this is just one-dimensional structure. One-dimensional structure. Now, second type of structure that is possible is secondary structure. Secondary structure means two-dimensional structure. Okay, two-dimensional. So what will happen? The primary structure was simply a thread-like structure. You can assume it as a thread. Okay. Now, if you coil the thread like this, if this thread is coiled or twisted like this in the form of a helix, if the thread is twisted in the form of a helix, or it is present in the form of sheet, beta sheet, then both these are examples of the secondary structure of the protein. Okay. So two types of secondary structures are possible. One is the helical form, okay, when the protein is present in the form of helix, or if the protein is present in the form of beta pleated sheet, okay, sheet like structure, it will be just like the sheet of paper. Okay, so these sheets of paper, you can see the sheets of protein are there, okay, beta pleated sheet. Both these are the secondary structures of the protein. Okay, now these sheets will be connected by the hydrogen bond. Okay, these will be connected together by the hydrogen bond. Here in the helix also, hydrogen bond will be present. So in the secondary structure, in helical structure as well as in the beta pleated sheet structure, hydrogen bonds will also be present in addition to the peptide bond. Okay, so in the secondary structure, hydrogen bond is also present along with the peptide bond. Okay, so this was secondary structure. Now tertiary structure. Tertiary structure. What is tertiary structure? It is a three-dimensional structure. Three-dimensional structure. So the primary structure was just a thread-like structure, long thread-like structure. In the secondary structure, that thread was coiled or twisted to form a helix, right? Now what will happen if the long thread is coiled upon itself or folded upon itself to form a complex structure just like a ball of wool? Have you seen a ball of wool? Okay, long wool is twisted or folded upon itself to form a complex ball-like structure. So the tertiary structure of the protein is similar to that. Okay, so it will be something like this, okay, folding of the thread-like structure to form a complex structure. Can you see this? So this is how the tertiary form of the protein will look like. Okay, this is the tertiary structure. Right. Now one important thing about the tertiary structure is that this is the biologically active structure. Biologically active structure. So which structure of the protein is biologically active? Tertiary structure. Therefore this tertiary structure is also acting like enzyme. Okay, so we will study enzyme after this protein. Okay, there we will see that enzyme is the tertiary structure of a protein. Okay. Here you can see that when you fold that thread-like structure to form a complex structure, certain pockets will be formed. Pockets or clefts or crevices will be formed in between. Okay, here you can see some pockets are formed. Okay, let me use some other color. Okay, so some pocket-like structures are formed here. Can you see? So some molecules can come and fit into these pockets. Okay, here also a pocket-like structure is there. Some molecules can fit themselves into these pockets, right? So these pockets or crevices are present in which structure of the protein? In the tertiary structure of the protein. Okay, so in case of enzyme, when the protein will be acting as an enzyme, then what will happen? The substrates will bind themselves at these pockets present in the enzyme. Okay, so one of the pockets will be acting like the active site of the enzyme. We will study that. Okay, now what is tertiary structure? So when any two of the different structures will combine together, then it will form tertiary structure. Okay, suppose the primary structure and the secondary structure combine together, it will form tertiary structure, or secondary or tertiary, sorry, it will form quaternary structure. If the secondary or tertiary structure will combine together, it can form quaternary structure. So quaternary structure is nothing but the combination of different structures of the protein. Okay. Quaternary structure is the combination of different structures of protein. One of the best examples of quaternary structure is the hemoglobin. Okay, we know that the hemoglobin consists of 4 chains. Hemoglobin consists of 4 chains. 2 alpha chains and 2 beta chains. Okay, therefore hemoglobin is a quaternary structure of the protein. Quaternary structure. Here you can see 2 alpha chains are there and 2 beta chains are there. So this is the quaternary structure of the hemoglobin. Okay, so I hope you have understood the primary, secondary, tertiary and quaternary structure of the protein, and you have also understood the peptide bond formation. Okay, which molecule is removed during peptide bond formation? Water molecule is removed. Therefore, this reaction is also a dehydration reaction. Now the next topic is enzymes. So what are enzymes? We have studied that the proteins can also be present in the form of enzymes. Okay, so most of the enzymes are proteins, but not all. There will always be some exceptions. Okay, so what is exception here? Sometimes RNA can also act as enzymes. Okay, so one of the example where RNA is acting as the enzyme is ribozyme. So ribozyme is a type of enzyme where RNA is present as the enzyme. Okay, it is formed from the RNA, nucleic acid, not the protein. Okay, but in general case or in most of the cases the enzyme will be protein only. Okay, therefore we are studying the enzyme just after the protein. Although in NCERT this topic is given at last, but here we are continuing this with the protein because most of them are proteins. Okay, then we have also studied that out of all the 4 types of structures of protein, that is primary structure, secondary structure, tertiary structure and quaternary structure, the biologically active structure was tertiary structure. Okay, so enzyme is the tertiary structure of the protein because tertiary structure is required for the enzyme to perform all the biological activities. Okay, and these enzymes differ from the inorganic catalysts. So what are enzymes? These are actually biocatalysts, right? Biocatalysts. That means they catalyze the biochemical reactions going on inside the body. Okay, then there are some inorganic catalysts also which catalyze the chemical reactions. So these enzymes are different from those inorganic catalysts. Okay, what is the difference between the inorganic catalyst and the biocatalyst, that is enzyme? In case of enzyme, that is biocatalyst, enzyme is a biocatalyst. This catalyst works efficiently at optimum temperature. Okay, so an optimum temperature is required to for the enzyme to work efficiently or productively. Okay, for example, the optimum temperature for human body is 37 degrees Celsius. Okay, for our body, human body, the optimum temperature is 37 degrees Celsius. Therefore, the enzymes which are working in our body, which are operating in our body, will also have this optimum temperature, this 37 degrees Celsius as the optimum temperature. So they will work most efficiently at this particular temperature. Okay, the temperature can be can slightly vary. It can be slightly more or less than 37 degrees Celsius, but it will be approximately here. Okay, it will be around 37 degrees Celsius. It can be 36, it can be 40 degrees Celsius. Okay, but it will not vary considerably. Okay, so the enzymes or the biocatalysts have some optimum temperature or optimum pH at which they will be most efficient. Okay, if you increase the temperature beyond that or decrease the temperature from below that, then they will not be active. Okay, below that temperature, below the optimum temperature, they will become inactive. And if you increase the temperature beyond that optimum temperature, then these enzymes can get damaged. Okay, they can get denatured. So beyond the optimum temperature, beyond optimum temperature, the enzyme will get denatured or damaged, and then what will happen? They will not be able to work again, okay, because these are finally destroyed. Okay, when they are denatured, that means they are finally destroyed, they cannot work again. But below the temperature, below the optimum temperature, they can become inactive. And if you increase the temperature slightly to their optimum temperature, they can again start working normally. Okay. So below the optimum temperature, they are not denatured, they are not finally destroyed, but beyond the temperature, they will get finally destroyed, and then they will not be able to work normally after that. Okay. Now there will be some exceptions. Okay, so here we have talked about the general human body temperature, that is 37 degrees Celsius. Now there can be some exceptional type of organisms which can thrive or which can tolerate the higher temperatures like 80 degrees Celsius to 100 degrees Celsius. For example, thermophiles. Example will be thermoacidophiles, right? Thermoacidophiles. Okay, so these are some of the bacteria which can live in the hot springs. Okay, they can tolerate the high temperature of the hot springs. So the enzymes which are operating in their body will also be able to work normally at this temperature. Okay, so for them, for the enzymes which are operating in the body of thermophiles, their optimum temperature will be around 80 degrees Celsius to 100 degrees Celsius. So this optimum temperature will vary from one organism to another. Okay. So this was about the biocatalyst, that is enzyme. Now how is it different from the inorganic catalyst? So inorganic catalyst, there is no such optimum temperature. Okay, they can tolerate different types of temperature. They can tolerate high temperature and high pressure. Okay, and it is generally believed that with every 10 degrees Celsius increase or decrease in temperature, the rate of the reaction can increase or decrease by 2 times. Okay, suppose the temperature is increased by 10 degrees Celsius. Okay, so there is 10 degrees Celsius increase in the temperature. Then what will happen? The rate of reaction will get doubled. Okay, it will become 2 times. Whereas if you decrease the temperature by 10 degrees Celsius, then what will happen? The rate of the reaction will become half. Okay, so the rate is becoming half or becoming double with decrease or increase in temperature by 10 degrees Celsius. So with every 10 degrees Celsius change in the temperature, the rate is changing. Okay. Now what is rate? I have said rate. I hope you understood what is rate. Rate is nothing but the amount of the reactant or substance consumed per unit time. This is the rate of the reaction. Or in terms of product also we can define the rate of the reaction. Okay, we can say that the rate is the amount of product formed per unit time. Okay, this is also defined as the rate of the reaction. For example, we have a reaction where we have 2 reactants A and B. So A and B are undergoing the reaction and they form a product C. Okay, so C is the product here, whereas A and B are the reactants. So how can you define the rate here? How can you write an equation to define the rate? So rate of the reaction can be written as minus dA by dt. That means the decrease in the amount of the reactant A per unit time. Okay, this is the rate of the reaction. Or you can also consider the reactant B and write that it is equal to minus dB by dt. Okay, the amount of decrease of the reactant B per unit time. Minus sign we have added because these are the reactants which are reducing, which are getting reduced with time. Okay, because product is being formed and the reactant is getting reduced. In terms of product, we can write dC by dt. Here we have not added minus sign. Here we can write plus sign, or simply we can not write anything. It will mean plus. Okay, because the product C is being formed, therefore its amount is increasing. So there is no minus sign. Whereas the amount of A and B are decreasing, therefore minus sign has been added. Okay, so this type of rate of the reaction can also be known as velocity of the reaction, because in this case the direction of the reaction is mentioned. Okay, so the direction is known. Direction of the reaction is known, whether something is decreasing or something is increasing. So here we can see that the reaction is going towards C, towards the product. Okay, so this is the rate, either the amount of decrease in the reactant per unit time or in the increase in the amount of product per unit time. This is known as the rate of the reaction. Okay. Now here you can see the enzyme activity. We know that the enzyme is a biocatalyst, so it will have some optimum pH and optimum temperature. So this is the pH, the top point. This is the optimum pH. Below this pH also it is decreasing, and above this pH also the rate is decreasing. Okay, enzyme activity is decreasing, that means the rate of the reaction is decreasing. Temperature also you can see there is an optimum temperature. The enzyme activity is the highest at this optimum temperature. Okay, so the enzyme will be most active at this point of temperature. Below this temperature, the activity is getting reduced, and above this temperature also the activity is decreasing. Okay, what will happen? Above the temperature, above the optimum temperature, the enzyme will get denatured or damaged permanently. Whereas below the optimum temperature, it will be temporarily inactive, which can get active again after it reaches the optimum temperature. Okay. Now we have to study about the reaction of the enzyme. Okay, how the enzyme reacts or how the enzymes work. So in order to understand the enzymatic reaction, first we should have an understanding about the general physical and chemical reactions. Okay, so the reactions can be of two types: physical reaction and chemical reactions. What is physical reaction? In physical reaction, no new product will be formed. Okay, the product will be same as the reactant. For example, if you take a paper, you take a sheet of paper and you tear these paper. Okay, so small fragments of paper are there. So although the paper has been torn into small pieces, but still each of the pieces is made up of only the same material. Okay, cellulose. Okay, paper consists of cellulose. Okay, it is made from the bark of the tree, right, which consists of cellulose. So these small fragments which are formed after tearing the paper, these are also containing the cellulose, right? They also contain cellulose. So is there any new chemical formed or any new product formed? No. Initially also it was cellulose, after the reaction also it is cellulose. Okay, so this is the example of physical reaction. Another important example of physical reaction is change in the state of matter. Okay, for example, we have solid water, that is ice. When you melt the ice, it will become water. Okay, so it can get converted into liquid, that is water. Similarly, if you boil water, this can get evaporated. Okay, then what will happen? It will form gas, that is water vapor. So this is also an example of physical change. There is only a change in the physical nature of water. The overall, the chemical nature of the water is same. Okay, we have H2O only. Only the bond between the water molecules has been loosened, right? But if you again condense the gas or the water vapor, it will again form the normal liquid water. Okay, if you freeze the liquid water, again it will again form the ice. Okay, so it can return in the same way. So no new chemical is formed. It's not that if you melt the solid ice, it will not form something else, like it will not form oil. It will form water only. Okay, so the nature, overall chemical nature is same, only the physical nature has changed. This is the physical reaction. Okay. But what is chemical reaction? In case of chemical reaction, new chemical will be formed. Okay, that will be entirely different from the reactant. Okay. Now chemical reaction can again be of two types. It can be inorganic reaction or organic reaction. Okay, inorganic reaction will include the inorganic molecules, whereas the organic reactions will include the organic molecules. So example of inorganic reaction is water molecule formation from the H plus ion and OH minus ions. Okay, so H plus ions and OH minus ions, these ions are different from the water that is formed after the reaction. Okay, here the chemical nature changes. Okay, similarly other example is, here we have barium hydroxide and this is sulfuric acid. So barium hydroxide is reacting with the sulfuric acid to form barium sulfate. So initially there was bond between barium and hydroxide, right, and hydrogen and sulfate. Now the bond has been formed between barium of one reactant and sulfate of another reactant. Now barium sulfate has been formed. Similarly water has been formed. So this barium sulfate is entirely different chemical from the barium hydroxide or sulfuric acid. Okay, so a new chemical has been formed. Therefore, this is the example of chemical reaction. Okay, so this was the example of inorganic chemical reaction. Similarly, we can see the example of organic chemical reaction as well. Best example of the organic chemical reaction can be the conversion of starch into glucose. Okay, when you eat something, let's say you eat a chapati, okay, you eat a bread that consists of starch. When you eat it, it gets digested and it will form glucose, which will give you energy. Okay, so how will the glucose give you energy? It can again undergo the glycolytic pathway. Glucose undergoes the glycolytic pathway, that is glycolysis. Again, we will be studying about the glycolysis in the chapter respiration that we are going to see very soon. Okay, after this chapter, we will have this chapter photosynthesis and then respiration. Okay, so in respiration, we will having a detailed look about the glycolysis, about the Krebs cycle. Okay, all these things will be discussed in detail in that chapter. So when the glucose undergoes the glycolytic pathway, it makes two molecules of pyruvate. Okay, also you should know that the glycolysis consists of 10 steps. Okay, glycolysis consists of 10 enzyme catalyzed steps. Enzyme catalyzed steps. Okay, so the glycolysis consists of 10 steps and at each step different enzymes are operating. Okay, so can you see the importance of enzyme? Now this pyruvate, two pyruvate molecules that are formed as a result of glycolysis can again undergo different reactions, okay, under different conditions. For example, under the anaerobic condition, that means in the absence of oxygen, they can undergo alcoholic fermentation and they can form ethanol, right? Then under the same anaerobic condition, that means in the absence of oxygen, if this reaction is taking place in the muscle of our body, then it can lead to the formation of lactic acid. So lactate fermentation can take place in the muscle of our body under the anaerobic condition. So both alcoholic fermentation and the lactic acid formation are taking place under anaerobic condition, that is in the absence of oxygen. Now what will happen if the oxygen is available, enough oxygen is available, then the condition will be aerobic condition. So what will happen under the aerobic condition? This pyruvate molecule will undergo the Krebs cycle and then electron transport system. Okay, so this is something that we are going to discuss in detail, so don't worry about this. If you don't understand this, we will discuss about this very soon. Here we have the example about the organic reactions. Okay, so you can see that the enzymes are taking part in the organic reactions. Okay, so I hope you have understood about the physical and the chemical reaction. This was given in the NCERT, so we have discussed this. Now, we know that the enzymes catalyze the reaction. Okay, catalyze the reaction. What do we mean by catalyzing the reaction? Catalyzing the reaction means increasing the speed of the reaction or increasing the rate of the reaction. Increasing the rate of the reaction. Okay. So here we have two examples which will show you how effective an enzyme can be. Okay, so we have the example when water is reacting with carbon dioxide to form H2CO3, that is carbonic acid. Okay. Now, if this reaction is taking place outside our body, suppose this reaction is taking place outside the body, then enzyme is not present. Okay, there is no enzyme. So the rate of the reaction will be normal. Okay, this reaction will take place at a normal rate. So as a result, what will happen? About 200 molecules of carbonic acid will be formed per hour. So in 1 hour, how many molecules of carbonic acid can be made? 200 carbonic acid molecules can be formed within an hour. Okay, in 1 hour. This is the normal rate of the reaction. But if the same reaction takes place inside our body with the help of the enzyme, that is carbonic anhydrase, okay, in our body we have an enzyme called carbonic anhydrase. Okay, this is an enzyme. So if this enzyme is present, then what will happen? It will increase the rate of the reaction to such a large extent that now the number of molecules of this carbonic acid formed will be about 6 lakh molecules per second, not per hour, per second. Okay, so initially in the absence of the enzyme, only 200 molecules were formed in 1 hour. Now in the presence of the enzyme carbonic anhydrase, about 6 lakh molecules are formed only in 1 second. So can you see that the presence of an enzyme has increased the rate of the reaction to about 1 crore times. Okay, so the effect of the enzyme is not small. Okay, the enzyme can be this effective. It can increase the rate of the reaction to lakhs of times or crores of times. Okay, so I hope you have understood the effectiveness of an enzyme. Now the next topic is enzyme activity. So we have seen that the enzyme is capable of bringing about a really high rate of chemical reaction. Okay, it can increase the rate of the reaction to many times, 1,000 times, lakhs of times, or even crores of times. But how can an enzyme do so? What is actually done by the enzyme? Okay, we have to find out. Now, if the question is simply asked what is actually done by the enzyme, and you have to answer in one line. So in one line the answer will be that the enzyme reduces or lowers the activation energy. So what is done by the enzyme? Lowering of activation energy. Lowering of activation energy is done by the enzyme. Now what is activation energy? Let's try to understand this with the help of the given graph. Okay, here you can see along the y-axis we have potential energy, or simply you can say energy, and along the x-axis we have progress of the reaction. Where is the substrate here? This is the substrate. And where is the product? This is the product. So can you see what is the difference between the substrate and the product? The substrate is at a higher level and the product is at a lower level. And what is that level? That is the energy level. Energy, potential energy. Okay. So the substrate is at a higher energy level and the product is at a lower energy level. Now this is not always the case. Okay. Sometimes the product can be at a higher level, but in this case the product is at a lower level and the substrate is at a higher level. Now the substrate has to get converted into the product. So what is required here? The energy of the substrate should become equal to the energy of the product. Okay, then the substrate will get converted into the product. So suppose this is point A, this is the energy level of the substrate, and this is point B, that is energy level of the product, right? Okay, this is point A, that is the energy level of the substrate. So in order to get converted into the product, the substrate has to come, the energy of the substrate has to come down and it has to become equal to the energy of the product. So can it come like this, directly from point A to point B? No, it is not the right mechanism. Okay, so the rule is that in order to come from point A to point B, first the energy of the substrate has to move to a higher level, like this, at this level. Let's say this is point C. Then first it has to reach at point C, and then from point C it has to come down to point B. This is the rule of the reaction. Okay, so before coming to point B, it has to come to point C, which is at a higher energy level than the energy of the substrate. Okay, and this point C is known as the transition state. So first the substrate has to reach to the transition state, and then from there it has to come down to the final level, that is the level of the product. Okay, therefore this is the shape of the graph that will be made. Okay, so here you can see this is the energy level of the substrate and this is the energy level of the transition state, right? Two dotted lines you can see. This is the energy level of the substrate and this is the energy level of the transition state. So the difference in these two levels is known as activation energy. So what is activation energy? It is the energy difference between the substrate and the transition state. Okay, the difference in the energy of the substrate and the energy of the transition state. The transition state will always be at higher energy level than the energy of the substrate. Okay, because this is the unstable state. Okay, there is high instability at this state. And how to understand this? Suppose this is the enzyme. And this is the substrate. Okay, now this substrate will come and it will fit into the enzyme. Okay, this is the enzyme. Now the substrate has fit into this enzyme. So when the substrate gets fit into the enzyme, there is an association between the substrate and the enzyme. This state is known as the transition state. Okay, this is known as the transition state. And this is highly unstable state. There is high instability because of having high energy level. Okay, this is a very high energy level state. So this is what is represented in this graph. Okay, so this transition state is nothing but the state in which the substrate and the enzymes are bound together or are associated together. Okay, so first the substrate and the enzyme has to come in contact. They have to bound together. They have to bind together, and then the substrate will get converted into the product and then it will be released from the enzyme, right? So this is the process which is shown through the graph. So this activation energy is very large, okay, very high energy. You can see there is a huge gap between the level of the substrate and the level of the transition state. So the substrate has to pass through this high energy, then only it will reach to the product level. So it will become very difficult for the substrate to pass through this energy level, and it will take a long time, right? It is similar to, let's imagine a situation, let's imagine a condition that the point A where the substrate is located, this is the home, this is your home, okay, and point B is your school. So you have to come from your home to your school. But the condition is that you have to take this pathway. Okay, this graphical pathway. First you have to reach to a transition state, and then from that transition state you have to reach to your school. So this is a long path you have to cover, so it will take more time and more effort, right? Now if I give you some shortcut, then what will happen? If I give you the shortcut, then you can reach your school in a very less time. Okay, so if earlier you were taking about 1 hour to reach the school, if I give you the shortcut way, then you can reach the school within 10 to 15 minutes. Okay, so this is what is done by the enzyme gives the shortcut way to the substrate, okay, by reducing the activation energy. So it will reduce the activation energy, or it will lower the energy, activation energy. So you can see in the red color graph, the red color line is there and pink color line is there. So red line shows the path of the substrate, path that the substrate has to go through without the enzyme, in the absence of the enzyme. But pink color shows you the path that the substrate has to go through in the presence of an enzyme. So in the presence of the enzyme, this path has been reduced because the activation energy has been reduced. So now it will easily pass through this way, and then it will reach the energy of the product. Okay, so this is what I meant by lowering the activation energy. Okay, I hope this is understood. Now two types of reaction can be there: exothermic and endothermic reaction. Exothermic means energy is released. Okay, exothermic means energy will be released, and endothermic means energy has to be absorbed. Energy absorbed. Now, in which case the energy will be released and in which case the energy will be absorbed? Let's see. In the first graph, this is the level of the substrate and this is the level of the product. So you can see this is the same graph as the earlier one. So here the product is at a lower level and the substrate is at a higher level. I am talking about the graph 1. This is graph 1, this is graph 2. So in the graph 1, the level of substrate is higher. So here, from here it will move to a transition state, and then from there it will move to the level of the product like this. Okay. Since the energy of the product is lower, that means the substrate has to release some energy in order to get to the energy level of the product. Okay, because it has to move from higher energy level to lower energy level. So energy will be released. Therefore, it is exothermic process or exothermic reaction. In the second case, the substrate is at a lower energy level and the product is at a higher energy level. So here the substrate will have to gain some energy or absorb some energy to reach to the level of the product like this. Okay, so here the energy has to be gained or absorbed. Therefore, this is endothermic reaction. Okay, so you have understood the exothermic and endothermic reaction. In exothermic reaction, the product will be at a lower energy level from the substrate, and in the endothermic reaction, the product will be at a higher energy level than the energy of the substrate. Got it? Now, what is the nature of enzyme action? Here you can imagine one thing, that enzyme is a teacher. Okay, it is just like a teacher, and the substrate is just like a student. Substrate is like a student. Student is preparing for NEET exam. Now it has to become a doctor. So student can do self-study also, right? But still students choose some teachers to guide them, right? If the student does not take help of any teacher and does only self-study, then it will require more effort and time because no guidance is available to the students or no direction is available to the student, right? But if the student comes in contact with a good teacher, then what will happen? Proper guidance will be provided. Okay, so it will take less effort and less time to succeed. Okay. So what happens? The enzyme is a teacher here. So suppose this yellow color is the enzyme, this is the teacher, and this green color substrate is the student. So you can see that the student came into contact with the enzyme, that is the teacher. Okay, this is the active site of the enzyme. The substrate will fit into the active site of the enzyme. Then what will happen? The teacher will provide guidance to the student. Then the student will get converted into the student will succeed in life with the help of the teacher and become a doctor. Similarly, the substrate with the help of the enzyme will become a product. Okay? Substrate will get converted into the product. Then after becoming doctor, do you still remain in touch with the teacher? Means you remain in touch but not daily, okay, just like you were in touch earlier. Okay, earlier you used to come to the teacher daily, you used to study from teacher, but now you don't have to do that. Okay, you move to your occupation, you become busy, and then you are busy in your work. Okay, then the teacher, what is teacher doing? Does teacher get converted into something? No, teacher does not get converted into something. Teacher remains a teacher. There is no change in teacher. After you move, then the next batch of students will come and the teacher is ready to teach another batch of the student. Similarly, substrate will get converted into the product, but there will be no change in the enzyme. Once this product is released from the enzyme, then the enzyme will be ready to bind a new substrate, right? So let me show you a diagram. So this is the enzyme, this is the substrate. The substrate comes in contact with the active site of the enzyme. Then what will happen? It will fit into the active site like this. The substrate has fit into the active site of the enzyme, so it becomes enzyme substrate complex. This is the enzyme substrate complex. Okay, now some bonds breaking and making will take place here and the substrate will get converted into the product. So this enzyme is there and now the substrate has got converted into the product. So this substrate enzyme complex will get converted into the enzyme product complex. Product complex. Now the work of the enzyme has been done, so the product can be released from here. The product will be released from here and now the enzyme will be ready to bind with another substrate molecule. Got it? So this is the mechanism of the enzyme action. Okay, this enzyme substrate complex is actually the transition state. Transition state, that is a high energy state. Energy is very high there. Okay, next is factors affecting enzymes. So we have already discussed earlier about the pH and temperature dependence of the enzyme. We know that there is an optimum pH and optimum temperature at which the enzyme will be most active. Okay, enzyme will be most efficient at that optimum pH or optimum temperature. So here in these two graphs you can see the highest point is the optimum pH and optimum temperature. Therefore, at this point the enzyme activity is highest. Now both below and beyond that optimum value the activity of the enzyme will decline. Okay, so below the optimum pH the enzyme activity will decrease and beyond this optimum pH also the enzyme activity will decrease. Similarly in temperature also, below that optimum temperature the enzyme activity will decline and the enzyme will become inactive, and beyond that optimum temperature the enzyme activity will decline and eventually the enzyme will become denatured or damaged permanently. Okay, this we have discussed. So I hope these two graphs are clear to you. Now the third graph is showing the dependence of enzyme activity on the substrate concentration or the relation between these two. So how is enzyme activity affected by the substrate concentration? Let's understand. Suppose we have 50 enzymes. We have 50 enzyme molecules, right? But the number of substrates is only 10. So these 10 substrates will bind to the enzymes. So only 10 of the enzymes will be active, okay, because only 10 substrates are there. So these 10 substrates will bind to 10 enzymes and 40 enzymes are not doing anything. So obviously the rate of the reaction will be lower. Now if the substrate concentration is increased, now we have 20 substrates. So these 20 substrates will bind to 20 enzyme molecules now. Now the rate of the reaction will increase because now more conversion of substrate into product is taking place. Then if the concentration of the substrate increases further, now we have 30 substrates. So these 30 substrates will bind to the enzyme, and this way the rate of the reaction will go on increasing, right? Or the velocity of the reaction will go on increasing. For 40 substrates, again it will increase further, and when the number of substrates become 50, then the velocity or the rate of the reaction will become maximum, because now we have 50 enzyme molecules and 50 substrate molecules equal. Now all the enzymes will be involved in the conversion of the substrate into the products. Okay, so all the 50 enzymes are doing the work, therefore the rate will be maximum. Now what will happen if the number of the substrates become more than 50? Let's say it becomes 60, 70 or 80. Will there be any change in the enzyme activity? No, because we have only 50 enzymes and all the 50 enzymes are already occupied now. So no more substrates can bind to the enzyme because no more enzyme molecules are available. So initially the rate of the reaction or the velocity of the reaction will increase as it is shown in this graph with the increase in the concentration of the substrate. But once it reaches a maximum value, then it will become constant. Okay, because now it has come to saturation level. When all the enzyme molecules are bound to the substrates and no more enzymes are available, then it is the saturation part, and then there is no change in the rate of the reaction or velocity of the reaction. So initially the rate will increase and then it will become constant after a certain point. Got it? So what is the difference between this third graph and the first two graphs? You can see that one thing that is common among all these three graphs is that before or below that optimum value, the rate of the reaction is increasing gradually in pH, in temperature, as well as in the substrate concentration. As the substrate concentration is increasing, then the rate of the reaction is also increasing. But once it reaches a particular point, then in case of pH and temperature, the rate starts decreasing, but in case of substrate concentration, it does not decrease, it becomes constant. Okay, so it will increase first and then it will become constant. Therefore, this type of graph is shown. Got it? So three factors are affecting the enzyme activity: pH, temperature and substrate concentration. Okay. Now what is Km? Here you can see this is the Km. This is a constant. And this is the substrate concentration at which the rate of the reaction or the velocity of the reaction is half of the maximum value. Okay, so if the maximum velocity of the reaction is Vmax, then here it is Vmax by 2. So the substrate concentration at which the rate is Vmax by 2, this substrate concentration is known as Km, which is a constant. Okay, so if the Km value will be lower, then it means that there is higher affinity between the enzyme and the substrate, and therefore the rate of the reaction will be faster, okay, or the reaction will be efficient. Okay, and if the Km value will be higher, then it means the reaction will be less efficient, okay, because it will mean that there is lesser affinity between the enzyme and the substrate molecules. Now we come to the enzyme inhibition. So the enzymes are actually very sensitive to some chemicals. Okay, enzymes are sensitive to some chemicals. Sensitive to some chemicals. So what will happen if these chemicals will bind to the enzyme? Then the enzyme activity will decline or the enzyme will get inhibited, because enzyme will not be able to function if these chemicals are bound with the enzyme. Okay. So this process is known as enzyme inhibition, inhibiting the activity of the enzyme. Now sometimes these chemicals resemble the substrate. They resemble the substrate. So here you can see we have this enzyme. This is the enzyme in this diagram. In white color we have the substrate. Okay, this is the substrate. So In normal case, this substrate has to come and bind to the active site of the enzyme, then the enzyme will catalyze the reaction. But what if there is a chemical, this is the blue colored chemical, to which the enzyme is sensitive, but this blue color chemical is resembling the substrate, okay, in the structure. You can see that these two, substrate and this chemical, are structurally similar. Okay, so the enzyme will get confused. It will not be able to identify which one is substrate and which one is the other chemical. So if the concentration of this chemical will be higher than the concentration of the substrate, then instead of the substrate, this chemical will bind to the enzyme, to the active site of the enzyme. Then what will happen? Will enzyme be able to function? No. The activity of the enzyme will get inhibited, and this is known as the competitive inhibition. Competitive inhibition. Because now this chemical or the inhibitor, this is known as inhibitor because it inhibits the activity of the enzyme, because the inhibitor will start competing with the substrate because it has same structure, similar structure, okay, and it is more in concentration also, so it will start competing with the substrate, and therefore it is known as competitive inhibition. Okay, one of the example is the enzyme succinate dehydrogenase. So this is an enzyme whose substrate is succinate, but sometimes malonate, which is structurally similar to the succinate, both have two carboxyl group, COOH. Here we have CH2, COOH. Other is COOH, CH2, CH2, COOH. So both are having two carboxyl groups. Okay, so structurally they are similar. They resemble each other, the succinate and the malonate. So actually the succinate has to bind to this enzyme succinate dehydrogenase, but sometimes malonate binds to this enzyme instead of the succinate. And then what happens? Eventually the enzyme gets inhibited. So this is one of the examples of competitive inhibition, because the malonate starts competing with the succinate. Got it? So this was about the enzyme inhibition. Now we come to the classification of the enzyme. So enzymes are classified into 6 classes. And then these classes are also further divided into subclasses, subclasses and subclasses. Okay, right. Now in order to name an enzyme, how is an enzyme named? So 4 digits are used to name an enzyme. 4 digits. Let's say 1, 2, 2, 1. Suppose, okay, it can be any digit, any number, but I am taking this example. So what is the first digit? The first digit represents the class of the enzyme. The second digit represents the subclass of the enzyme. The third digit represents the subclass. And the fourth digit represents specific number or code given to the enzyme. Okay, so we don't have to go into detail, you just have to remember this rule. We just have to discuss the class. We don't have to go into detail about the subclass and subclass. For example, here you can see we have to study about 6 classes because there are 6 classes of enzyme. So if we are naming an enzyme which belongs to the first class, then the first digit will be 1. If it belongs to the second class, that is transferase, then the first digit will be 2. Similarly, it can be 3, 4, 5 or 6 depending on which class the enzyme belongs to. Similarly, the subclass and subclasses will also be mentioned. Okay. So now let's discuss all the 6 classes of the enzymes. In order to remember the names or the classes, you can remember this OTHLIL, OTHLIL, where O represents the first class of the enzyme, that is oxidoreductase. T represents the second class of the enzyme, that is transferase. H represents the third class of the enzyme, that is hydrolase. L represents the fourth class, that is lyase. I represents the fifth class, that is isomerase. And the L at last represents the sixth class of the enzyme, that is ligase. So by remembering this oath lil, you can remember the sequence of these enzymes, okay? Because you have to remember the sequence, which is the first class, which is the second class, which is the third class. So total 6 classes are there. The first class is oxidoreductase or dehydrogenase. So what do we mean by oxidoreductase? Oxido means oxidation, reductase reduction. So this enzyme will be involved in catalyzing the oxidation plus reduction reaction, that means redox reaction. So the oxidoreductase or dehydrogenase enzymes will be involved in catalyzing the redox reaction. So one substance will get reduced, other substance will get oxidized. So here is the example. Suppose we have the substrate S that is reduced and the substance 2 S dash that is oxidized. So this enzyme will help in catalyzing the redox reaction. So now after the reaction, what will happen? S, which was initially reduced, will become oxidized now, and S dash, which was initially oxidized, will now get reduced. Okay, so the redox reaction has taken place. Okay, so oxidoreductase enzymes will be involved in this type of reaction. Second class is transferase. Now, as it is clear from the name, it will help in transferring of the groups. Transfer of the groups. So it is involved in catalyzing the transfer of a group other than hydrogen between a pair of the substrates. So we have two substrates. We are taking two types of substrates. One is S and the other is S dash. Okay. So why we have written other than hydrogen? Because the transfer of hydrogen is already involved in the redox reaction. Okay, so if the transfer of hydrogen is taking place, it will be part of the redox reaction, not this one. Okay, other than hydrogen, if there are other groups, so this is S substrate, this is bound to a group G. Okay, G represents a certain group, and S dash is not bound with anything. Now this enzyme will transfer this group from S to S dash. So after this reaction, you can see that S is isolated and the group is bound with the S dash. Okay, so there is a transfer of the group from one substrate to another substrate, and the enzyme involved will belong to the class transferase. Then the third class is hydrolase. So again, it is clear from the name, you can understand from the name only. Hydrolase means it will be involved in the hydrolysis. So hydrolysis of ester, ether, peptide or glycosidic bonds, etc. Okay, hydrolysis means what? Breaking of the bonds. Breaking of the bonds with the help of water. So water will be involved here in hydrolysis. Then lyase. Lyase enzymes are also involved in breaking the bonds. These are also involved in breaking bonds, but this time water will not be involved. Okay, so it will be without water. Without water. So the lyases are involved in catalysis of removal of groups from the substrates by mechanism other than the hydrolysis, leaving the double bonds. Okay, so it will also involve the removal of groups, that is breaking of the bonds, but it will not include the hydrolysis. Okay, it will not include the water because that will be part of the hydrolase. Okay, that will be done by the hydrolase. So example is shown here. We have this substrate where carbon-carbon single bond is there, then one carbon is bound to X, the other carbon is bound to group Y. Okay, now these groups X and Y are removed from here by the enzyme lyase. Okay, and this will lead to a double bond between these two carbons. Okay, so eventually there will be double bond between these two carbons and groups XY will be removed from the carbons. Which class of the enzyme will be involved here? The lyase class. Okay. Then the fifth class is the isomerases. Again, it is clear from the name, it will be involved in the catalysis of the conversion of one isomer into another isomer. So interconversion of optical, geometrical or positional isomers. Okay, they will be involved in the conversion of one isomer into another. And the last and the sixth class is the ligase. There are two similar names, lyases and ligases. So do not get confused between these two. Lyases are used in breaking the bond or removal of the groups. Removal of the groups. Whereas ligases are involved in making of the bonds or linking of the group. Linking of the group. Okay, so they have opposite works or opposite functions. So ligases are involved in linking together of the two compounds. Okay, like carbon oxygen, carbon sulfur, they can form bonds. They will be involved in forming the bonds, not breaking the bonds. Okay. Next topic is cofactors. So what are cofactors? See, the suffix co means with or together, right? It means with or together. So these are the factors which are associated with the enzymes. We know that the enzymes are proteinaceous in nature. Okay, most of the time enzymes are protein. Enzymes are protein in nature, right? So some of these proteins are not capable of catalyzing a reaction alone. Instead, they need the assistance of some non-protein factors. So when these proteins get combined with the non-protein factors that are also known as cofactors, then they are able to catalyze the reaction. Okay, so enzymes are protein part, whereas the cofactors are non-protein part. Okay, and the protein part is known as apoenzyme. Apoenzyme. Okay, and together they are known as holoenzyme. Okay, so without the cofactor, these enzymes will not be able to catalyze the reaction. So what are cofactors? These are the additional factors, additional non-protein factors which are associated with some of the enzymes. Okay, and without these cofactors, these enzymes will get inactivated. They will get activated only if they are combined with these cofactors. Okay, there are three types of cofactors. First one is prosthetic group, second one is coenzyme, and the third one is metal ions. Out of these three, both prosthetic group and the coenzymes are organic in nature, whereas the metal ions are definitely inorganic, right? It is already written here. The prosthetic group is organic, coenzymes are also organic, but the metal ions are inorganic. Okay. Now, when prosthetic groups and coenzymes both are organic molecules, then what is the difference between them? So the difference is that the prosthetic groups bind tightly with the enzyme. They bind tightly with the enzyme, whereas the coenzymes are bound loosely with the enzyme. Okay, so they have a transient or loose association with the enzyme. Okay, prosthetic groups are bound tightly and the coenzymes are bound lightly or loosely. Okay, but both are organic molecules. There are examples of the prosthetic group. Here we can see the reaction. Hydrogen peroxide is giving water and oxygen. Okay, so there is the breaking of hydrogen peroxide molecule and water and oxygen are released. In this reaction, the enzyme that is used is peroxidase catalase. But this is the enzyme. Now this enzyme peroxidase catalase will be inactivated without a cofactor. It will get activated only if a cofactor, that is prosthetic group, will get attached to it. And what is that prosthetic group? Heme. Okay, heme is the prosthetic group which is required by this enzyme peroxidase catalase to catalyze this reaction. In absence of the heme group, it will not be able to catalyze this reaction. Okay. Now let's see the coenzyme. So we know that this is the organic again. Now it has loose association or transient association with the enzyme. These are usually vitamins. Okay, coenzymes are usually vitamins. Therefore, we say that we should take vitamins and minerals in our diets. Why? Because vitamins act as the coenzymes in our body for many reactions. And similarly, minerals are types of metal ions. Okay. The example of coenzymes are NAD, NAD and NADP. What is the full form of NAD? Nicotinamide adenine dinucleotide. And NADP means nicotinamide adenine dinucleotide phosphate. Okay, P for phosphate. And these as I said that these coenzymes are most of the time vitamins. So NAD and NADP, these are derived from which vitamin? From the vitamin niacin, that is vitamin B3. Okay, these are the derivatives of vitamin B3. Now the third type of the cofactor is metal ions, which are inorganic obviously. These form coordinate bonds. So the metal ions form coordinate bonds with the enzyme as well as with the substrate. So these metal ions act as the bridge between the enzyme and the substrate. Okay, I hope the screen is clear to you. Let me scroll it up. Example of metal ion is zinc ion, Zn2 plus in alcohol dehydrogenase or carboxypeptidase. Okay, so alcohol dehydrogenase or carboxypeptidase, these are the enzymes which need the metal ion, that is zinc ion. Okay, so suppose this is the enzyme and this is the substrate. Okay, this round one is the substrate. Now what will zinc ion do? Zinc ion will make a bridge between these two. So this will form the bond with this substrate as well as with the enzyme. Okay, so which type of bond is formed by the metal ions? Coordinate bonds. Now here we have a diagram showing the relation between the enzyme and the cofactor. Here you can see the first one is an enzyme. So we know that the enzyme is proteinaceous. So when the protein and the non-protein cofactors are combined, the protein part will be known as apoenzyme. Okay, so this protein part is the apoenzyme, whereas the non-protein part is the cofactor, which can be a prosthetic group, a coenzyme, or a metal ion. Okay. Here you can see without the coenzyme, this enzyme is inactive. But when it will get attached to this cofactor, then it will get activated and now it will be able to bind with the substrate. Okay, without the cofactor, it will not bind with the substrate. So it cannot do anything without the cofactor. This is not true for all the enzymes, only some of the enzymes. Okay, most of the enzymes can do work by themselves. Now the next topic is lipid. We have studied about the proteins. Proteins are a type of biomacromolecules. Enzymes are a type of proteins. Okay, enzymes are the biocatalysts, which are type of proteins, most of them. Now we come to another type of macromolecules, that is lipids. Now we had studied that the lipids, their weight, their molecular weight was less than 1000 Dalton. Right? So according to weight, they can be called as micromolecules, but since they were found among the acid insoluble pool, among the macromolecules, therefore these are also placed, these lipids are also placed among the biomacromolecules. Okay, so when we study the lipids, we generally study them under the macromolecules. But if the question is asked particularly according to the molecular weight, then according to the weight they can be placed under the micromolecules. Okay, now these lipids are mostly water insoluble because these are hydrophobic. Lipids are hydrophobic. Okay, now most of the lipids are made up of fatty acids. Okay, fatty acids are the building blocks of the lipids. Building blocks of the lipids. Just like the amino acids were the building blocks of the proteins, right? But here, not all the lipids are made up of fatty acids, but many of them are made up of fatty acids. Therefore, we study the fatty acids. Now, what are fatty acids? So these are fat, or we can say they are long hydrocarbon chain with a carboxyl group. So the fatty acid will consist of a hydrocarbon chain or an alkyl group, right? And there will be a carboxyl group attached to it, like this. Okay, this is the fatty acid. A long hydrocarbon chain will be there and a carboxyl group will be attached to it. Now the number of carbons can vary in that hydrocarbon chain. It can be 15 carbon, 16 carbon, 20 carbon. Any number of carbon can be present. Okay, examples are arachidonic acid. So arachidonic acid are a type of fatty acid which consists of 20 carbons. 19 carbons are present in this hydrocarbon chain and 1 carbon is part of carboxyl group. So 19 plus 1, 20. So arachidonic acid consists of 20 carbons. Similarly, for palmitic acid consists of 16 carbons. So in the hydrocarbon chain, 15 carbons will be present and 1 carbon will be part of the carboxyl group. So total 15 plus 1, 16. Okay, palmitic acids are made up of 16 carbon and the arachidonic acid are made up of 20 carbon. So there are many different types of fatty acids depending on the number of carbons that are present in them. Now these lipids can be saturated or unsaturated. What is meant by saturated? Saturated means there will be no double or triple bonds. Okay, all bonds will be single or sigma bonds will be present. Okay, so single bonds. Whereas unsaturated lipids can have double bonds. I hope the basic things are clear. What is saturated or unsaturated? Okay, saturated does not contain any double bond, but the unsaturated can contain double bond. They will contain the double bond. Here we have the example. You can see oleic acid. The first one is the oleic acid. You can see one double bond is there, right? Whereas in palmitic acid, can you see any double bond? Here you don't have to count the double bond of the carboxyl group. Okay, carboxyl group is different. Here we have to check whether the hydrocarbon chain has any double bond. If this is the hydrocarbon chain, is there any double bond in this hydrocarbon chain? We don't have to see the carboxyl group. So in the oleic acid, there is one double bond. So this is unsaturated. Okay, this is the example of unsaturated. Whereas palmitic acid, there is no double bond in the hydrocarbon chain. Therefore, this is saturated lipid. Saturated lipid. Again, in the arachidonic acid, you can see we have discussed that the arachidonic acid consists of 20 carbons. So you can count the number of carbons: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19 and 20. So 20 carbons are present, including the carboxyl group. Okay. Now, you can see there are numerous double bonds in this arachidonic acid hydrocarbon chain. Okay, you can see so many double bonds: 1 double bond, 2, 3, 4. 4 double bonds are present in the hydrocarbon chain of the arachidonic acid. So this is also which type of lipid, saturated or unsaturated? Again, it will be unsaturated. So whenever double bonds will be found in the hydrocarbon chain of the fatty acid, then it will be unsaturated. Okay. Now, you can think about one thing. Suppose this is your saturated fatty acid, okay, because there are no double bonds. So we know that the lipids are made up of the fatty acids, okay. Fatty acids are the building blocks of the lipids. So if we have to make a lipid, then a large number of such fatty acids will be present, right? So this is one fatty acid, then there can be another fatty acid like this, then another fatty acid. So a large number of fatty acid together will form the lipid. Okay, and here all the fatty acids are saturated fatty acids. They do not have any double bond. So you can see that the packaging will be very tight here. Okay, they will have high packing efficiency. High packing efficiency. So when there will be high packing efficiency, their arrangement will be tight. Okay, they will be tightly arranged with each other. Therefore, they will have high melting point. High melting point, and therefore they will behave like solid at room temperature. So they will remain as solid at room temperature. The example of such lipids are coconut oil. Okay, coconut oil. Coconut oil. Then another example can be ghee. So these things remain solid at room temperature. Why? Because they have saturated fatty acids in them. They are made up of saturated fatty acids. There are no double bonds. So they are tightly packed with each other. Okay. On the other hand, if a lipid is made up of unsaturated fatty acids, then what will happen? Let's see. Suppose this is one fatty acid, this is unsaturated. So we have a double bond here. Okay, here we will have the carboxyl group. Then we have another fatty acid. Again, due to double bond, there will be some distance between them. Okay, another fatty acid. So the double bond will lead to distance between two fatty acid molecules, right? So this distance will lead to loose arrangement, okay, loose arrangement. Or we can say that they will have less packing efficiency or low packing efficiency. Low packing efficiency. So they will remain a solid or liquid at room temperature. They will remain a liquid at room temperature. Okay, they will remain like a liquid at room temperature. Room temperature. Because they will have low melting point. Low melting point. So these are loosely arranged because of the presence of the double bond. So they will have low melting point. They can melt very easily at very lower temperature, right? So they will remain as a liquid in the room temperature. On the other hand, the lipids which are made up of saturated fatty acids will remain like a solid at room temperature because they will have high melting point. Okay. What will be the example of these lipids? The oils, mustard oil, right? Mustard oil will be the example. So any oil that remains a liquid at room temperature will be made up of the unsaturated fatty acids, whereas the oils which can solidify in room temperature, then they are made up of the saturated fatty acids. Okay, so this you have to understand. Now, fats and oils are the most common example of lipids. Lipids can be of different types. Fats and oils are not the only example of lipids. There are other examples as well, but these are the most common examples, right? Because these are commonly found in our home. We use them commonly, right? So how are fats and oils made? The basic structure of the fats and oils are same, right? The only difference between the fats and oils will be that the fats will be made up of saturated fatty acids. They will be tightly packed, therefore they will have high melting point, whereas the oils will have unsaturated fatty acids, so they will have low melting point. Okay, the only difference is this in the type of fatty acids, but the overall structure of fats and oils will be same. So what is the structure? Fats and oils are made up of glycerol and fatty acids. Okay, they are made up of glycerol and fatty acids. Glycerol is an alcohol, right? Glycerol is an alcohol, whereas fatty acids are acid, right? So here the reaction between alcohol and an acid is taking place. Okay, this is a reaction between alcohol and acid. So this type of reaction is known as esterification. Esterification. And the bond that is formed between the alcohol and the acid is known as ester bond. Ester bond. So which type of bond will be found in the fats and oils? Ester bond will be found. Okay. Now what is glycerol? Glycerol is trihydroxypropane. Another name of glycerol is trihydroxypropane. Trihydroxypropane. That means if you have propane consisting of 3 carbons and each of the carbons have a hydroxyl group, okay, that means in total 3 hydroxyl groups are attached to all the 3 carbons of the propane, then this will be known as trihydroxypropane, or in short they are known as glycerol. Okay, so glycerol is must. Glycerol has to be present in fats and oils. In addition to the glycerol, there will be fatty acids. Okay, now the fatty acids can be different to all the 3 parts, to all the 3 hydroxyl groups. Different fatty acids can be attached. Okay, so here you can see 3 fatty acids are there. This is the glycerol. This is the glycerol here. Here we have 3 fatty acids. This one. Fatty acid consists of one alkyl group or a hydrocarbon chain and carboxyl group. So this is the carboxyl group of the fatty acid and this is the alkyl part. Similarly, we have second fatty acid and third fatty acids. So 3 fatty acids are getting attached to the 3 positions of the glycerol. Okay. So here you can see the reaction that is taking place. You should understand this reaction well. Let me zoom in. Okay. So can you see this reaction? In the lowest most carbon, this reaction is taking place. So this is the fatty acid on the right side, and on the left side we have the last carbon of the glycerol. So this OH of the glycerol is reacting with the OH of the carboxyl group of fatty acid. Now this OH of the fatty acid will be removed from here, and the only hydrogen of the glycerol is removed from here. So H2O will be removed. Okay, water molecule will be removed and a bond will be formed between the two. So only oxygen will be common. This type of bond will be created. Okay, similarly the bonds will be created on other carbons also. So three carbons are present in the glycerol. These are reacting with 3 fatty acids. So 3 ester bonds are formed. Okay, so this is the structure of the fat. Okay, and while making each bond, 1 water molecule is removed. So while making 3 bonds, 3 water molecules will be removed. Okay, so I hope you have understood this. Now these R1, R2 and R3 can be different, right? If it is fat, then these alkyl groups will be saturated. Okay, these hydrocarbon chains will be saturated. If it is oils, they will be unsaturated. Okay, so the only difference between the fats and oils is in the type of the fatty acids. Okay, but the overall structure will be same. Now, it is not compulsory that all the 3 hydroxyl group of the glycerol will be combined with the fatty acid. If only one bond formation take place, that means only one fatty acid is present in the glycerol, then it is known as monoglyceride. If 2 fatty acids are present, are combined with the glycerol, then it is known as diglyceride. If 3 fatty acids react with the glycerol, then it is known as triglyceride. Okay, so depending on the number of the fatty acids to which the glycerol react, it can be monoglyceride, diglyceride or triglyceride, but 3 is the maximum number. Okay, it cannot be tetraglyceride because in glycerol we have only 3 carbons, so maximum 3 fatty acids can be attached to them. Okay, now the lipids are of 3 types. They can be of 3 different types: simple lipids, conjugated lipids or derived lipids. Simple lipids are the fats and oils that we have discussed in detail. We have already seen the structure of fats and oils. These are the simple lipids. So all the structures that we discussed right now were the structure of fat and oil. These are type of simple lipids. Another type of lipid is conjugated lipid, which are also known as complex lipid. Why complex? Because the overall structure of the conjugated lipid will be same as the simple lipid. That means glycerol will be present. Glycerol plus fatty acids will be present. But in addition to glycerol and fatty acids, conjugated lipids can also have some different groups attached to them. So they can have additional group other than the glycerol and fatty acids. For example, phosphate group. Okay. Example of the conjugated lipid is lecithin, which is found in the cell membrane. Lecithin is found in the cell membrane of our cell. Okay, here is the structure of the lecithin. Okay, this is the structure of the lecithin, and if you want, you can remember the structure of the lecithin because I think it is given in the NCERT. Then the third type of lipid is derived lipid. So what are derived lipids? These are the lipids which are derived from the simple or conjugated lipids. Okay, sterols are types of derived lipids. For example, cholesterol. Here we have the structure of the cholesterol. Again, these two structures, lecithin and the cholesterol, both are quite complex. But if you want, you can remember this. You can use some tricks to remember the structures. Okay, because these can be asked in the examination. So the lipid is completed. Now we come to the next topic, that is polysaccharide. Polysaccharides are another type of biomacromolecules. These are also known as carbohydrate. So why these are macromolecules? Again, because they were found among the acid insoluble pool in that experiment that we had discussed earlier. So they were found in the acid insoluble pool. Therefore, these are called macromolecules. And the building blocks of the polysaccharides are monosaccharides. Building blocks of the polysaccharides are monosaccharides or sugars. Monosaccharides or sugars. Okay, monosaccharides are also known as sugars. What are monosaccharides? These are polyhydroxyaldehyde or polyhydroxyketone having at least one chiral carbon. Okay, so the building blocks of the polysaccharides are monosaccharides. Similar to the building blocks of the proteins were amino acids, right? Building blocks of the lipids were fatty acids. Similar to that, the building blocks of polysaccharides are the monosaccharides or sugars. So what are monosaccharides? Monosaccharides can be aldehyde or ketone, and they are polyhydroxyaldehyde or polyhydroxyketone. That means a large number of hydroxyl group, OH groups, will be attached to them. Okay, and they must have at least one chiral carbon. What do we mean by chiral carbon? We know that a carbon can be attached to 4 different groups, right? Because the valency of the carbon is 4. So if all the 4 groups attached to the carbon are different, G1, G2, G3 and G4, then this carbon is known as chiral carbon. Okay. So all the 4 groups must be different in a chiral carbon. Okay, so in a polyhydroxyaldehyde or polyhydroxyketone, that is in the monosaccharide, at least one chiral carbon must be present. So we have the example here: glucose, fructose, galactose. These are the examples of monosaccharides. Okay, so you can see the structure of glucose and fructose. Both glucose and fructose are 6 carbon molecules. 6 carbon molecules. The only difference between glucose and fructose is that glucose is an aldehyde, polyhydroxyaldehyde, whereas fructose is a ketone, polyhydroxyketone. Okay, so you can see in the glucose we have this carboxyl group, this aldehydic group, and here we can see the ketone group shown in the pink color. Okay, other you can see total how many carbons are there? 1, 2, 3, 4, 5 and 6. 6 carbons are there, right? And polyhydroxy, you can see a large number of OH groups are there. 1, 2, 3, 4, 5. Okay. Similarly, you can see the structure of fructose. The only difference is that the glucose is aldehyde, so it consists of an aldehydic group, and the fructose is ketone, so it will consist of ketone group. Okay, and the ketone part will be on the second carbon, not on the terminal carbon. Okay. So this was about the monosaccharides. Now what are oligosaccharides? The oligosaccharides consist of 3 to 10 monosaccharides. Okay, so if 3 to 10 monosaccharides are arranged together or these are combined together, they form the oligosaccharides. Okay, you can also call them polysaccharides, but if the number is few, then they are mostly called oligosaccharides. If they are more than 10, then they will be known as polysaccharides. Okay, so 3 to 10, actually it is 2 to 10. Okay, 2 to 10. Because even if 2 monosaccharides are there, it will be called as oligosaccharides. Only if 1 monosaccharide is there, that is just monosaccharide. So the examples of oligosaccharides, you can see sucrose and lactose. Sucrose consists of 2 types of monosaccharides, glucose and fructose. Okay, so sucrose is made up of glucose and fructose, whereas lactose is made up of glucose and galactose. So here we have glucose and galactose. Okay, so these are oligosaccharides. Now polysaccharides, if the number of monosaccharides is more than 10, then it will be known as polysaccharides. Now we have to see the structure of glucose and ribose. So let's see the structure of glucose. We have already seen the structure of glucose there along with the structure of fructose. So we know that total 6 carbons will be there. 1 carbon, then carbon number 2, carbon number 3, carbon number 4, carbon number 5, and then this is carbon number 6. 6 carbons are there. Okay, this is OH group on the right side, then left side, then right side, right side. This is the structure that you should remember. Okay, you will have to remember the proper structure. You cannot change the position of hydrogen and hydroxyl group. This is the structure you have to remember. Then the structure of the glucose can be written in the linear form as well as in the cyclic form. So this is the cyclic form of the glucose. Okay, you can see that all the OH which are present on the right side in the linear structure, in the cyclic structure they are located on the lower side. Okay, so in this CHO aldehyde group, here you can see OH group, this is on the lower side here. Then this second OH group again lower side because it is on the right side here. Then third one is on the left side, so this is written on the upper side. Okay, again fourth one and fifth one on the lower side. Okay, right. So this is how you can remember the structure of the glucose. Similarly, we have the structure of ribose. So what is the difference between glucose and ribose? Glucose is a 6 carbon molecule, but ribose is a 5 carbon molecule. Okay, 5 carbon molecule. Here you can see this is carbon number 1, carbon number 2, carbon 3, 4 and 5. So only 5 carbon molecules will be present. Carbon will be present in the ribose. Okay. Here OH is present on the same side. All the 3 hydroxyl group are present on the same side here, unlike the glucose. So the structure of ribose is simpler than the structure of the glucose. So since all the hydroxyl group are on the right side, therefore here also you can see all the hydroxyl group on the lower side. Okay, so these are the structures of linear structure and cyclic structure of the ribose. This is the cyclic structure of ribose that we have seen just now. We have this OH. Now if you start from this first, number 1, then number 2, suppose this is 5 carbon molecule, right, ribose. So this is carbon number 1, this is carbon number 2, 3, 4. Okay, so from the carbon 2, from the second position, we have the OH group. So if we remove this oxygen from the second position, or when we delete this oxygen, delete the oxygen from second position, then it will be known as deoxyribose. That means oxygen has been deleted from here. So you can see it is shown in the pink color. In ribose we have OH, whereas in deoxyribose we have only H because oxygen has been deleted or removed from here. Okay. So this you will see in the nucleic acids because the DNA consists of deoxyribose. Now let's summarize the polysaccharides, the types of polysaccharides which are mentioned in the NCERT. So we have seen that the polysaccharides can be homopolymers or heteropolymers. So they can be homopolysaccharides or heteropolysaccharides. That means homopolysaccharides means what? All the monosaccharide units will be same. But in heteropolysaccharides, all the monosaccharide units can be different. Okay. So the examples of polysaccharides. Let's first discuss the homopolysaccharides. The examples are cellulose. Cellulose is a homopolysaccharide which is formed by the repeating unit of glucose. So they will have only one type of monosaccharide, and that is glucose in case of cellulose. Okay, and cellulose is found in the plant cell wall. The paper that we see, the cotton fiber, all these are example of cellulose, right? Then starch is also homopolysaccharide, right? It is actually a variant of the cellulose. It is variant of the cellulose, and it also consists of glucose because cellulose is also made up of glucose only. And the starch acts as the storehouse of energy in plants. So this works as for storage in plants. The starch can be present in 2 forms, amylose as well as amylopectin. Amylose is the unbranched form, whereas amylopectin is the branched form. Then third type of homopolysaccharide is glycogen. It is another variant of cellulose which is used in storing the energy in case of animals. So starch stores the energy in the plant, and the glycogen stores the energy in animals. So the only difference between starch and glycogen is that starch is present in plants and glycogen is present in animals or including humans. Okay, so this is also made up of glucose only. The function of both starch and glycogen is storage. Only difference is that starch is in plants and the glycogen is in animals. Then another example of homopolysaccharide is chitin. Now this is made up of a complex sugar called N-acetyl glucosamine. Okay, N-acetyl glucosamine. In short, we call it NAG. Okay, so whenever we will use this NAG, then you will understand that we are talking about N-acetyl glucosamine. This chitin is found in the cell wall of fungi as well as in the cytoskeleton of the arthropods. Then the last example of homopolysaccharide is inulin, which is made up of repeating unit of fructose. So we have seen starch, cellulose, glycogen, all these were formed by the repeating unit of glucose, but inulin is formed by the repeating unit of fructose. So here the monosaccharide is fructose. Okay. So these were the different types of homopolysaccharides. Now we come to the heteropolysaccharide. They will have different monosaccharide units. Example is peptidoglycan, which is found in the cell wall of bacteria, right? It is made up of two types of monosaccharides. First one is NAG, we have discussed right now, N-acetyl glucosamine. NAG is the N-acetyl glucosamine that we have seen here, plus NAM, that is N-acetyl muramic acid. So peptidoglycan is made up of two types of monosaccharides, N-acetyl glucosamine and N-acetyl muramic acid. Similarly, hyaluronic acid, this is also made up of two types of monosaccharides. First one is N-acetyl glucosamine and the other one is glucuronic acid. Okay, so these are some difficult names that you should remember. So we have seen most of the things about the polysaccharides, only some structures are left. So this is the structure of glycogen, okay, glycogen. Now from this figure you should notice few things. Just have a look at this position. Here you can see, we know that the glycogen is made up of repeating units of glucose because it is a homopolysaccharide. So it will consist of only one type of monosaccharide, that is glucose. So there is no doubt that only glucose is present in this. Okay, you can see this is a branched molecule, glycogen is branched. So you can see that in this glucose, suppose this is one glucose. Glucose is a 6 carbon molecule, right? So this is 1, 2, 3, 4, 5, 6. Here we have oxygen. Again, we have this another 1, 2, 3, 4, 5, 6. Okay, so these are two glucose molecules that have to combine together. The bonds which are present between two monosaccharide units are known as glycosidic bonds. So these two glucose have to make glycosidic bonds. So you can see this is carbon number 1, 2, sorry. We start from here, right? So 1, 2, 3 and 4. Similarly, here we have carbon number 1, 2, 3, 4. Now you can see that the bond, glycosidic bond, is formed between this carbon and this carbon. That means between carbon number 1 of one glucose and carbon number 4 of another glucose. So this glycosidic bond is 1,4 glycosidic bond. That means it is formed between carbon number 1 of one glucose and carbon number 4 of another glucose. Okay. So this you have to remember. But this is about the linear part of the glycogen. This is the linear part where all the glucose molecules are attached in a linear form. But this glycogen also has branching. So at some position branching will take place. So this is the linear part here. This is another linear part. But at this position branching is taking place, right? At this position branching is taking place. So between the branches you can see that here this is carbon number 1 but this is carbon number 6. Okay, so the branching will take place between carbon 1 of one glucose and carbon 6 of another glucose. Okay, so in the linear part, the glycosidic bond formation will take place between carbon 1 and carbon 4. Whereas at the point of branching, the bond formation will take place between carbon 1 and carbon 6. Okay, carbon 1 of one glucose molecule and carbon 6 of another glucose molecule. So this difference you have to remember. Now one thing that we have missed out about starch is that starch is a helical structure. Okay, the structure of starch is helical like this, whereas simple cellulose is a linear structure. Okay, so the starch has a helical structure whereas cellulose is a linear structure. So since the starch has helical structure, it has some position. You can see these locations at the point where helix are forming. Therefore, starch gives iodine test. Iodine test. Because these locations can hold iodine molecules. Okay, iodine molecules can be held at these locations where helices are being formed. Whereas in case of cellulose, there is no such helix, only linear structure is there. So it cannot form iodine and it cannot hold iodine molecules. Okay, cannot hold iodine molecules. Therefore, it will not give the iodine test. Okay, only starch will give the iodine test. So it will give blue-black color of iodine. Got it? So this also you have to remember. Starch gives the iodine test because it has helical structure. So it can hold the iodine molecules. Whereas the cellulose has linear structure. It cannot hold iodine molecules. So it will not give the iodine test. Now these are some of the structures that were given in the NCERT. You can see sugars, that is carbohydrates. So first one is glucose. We have seen the structure of glucose. Another is ribose. Glucose is a 6-carbon molecule. Ribose is a 5-carbon molecule. Then these are the structures of some amino acids. This is glycine, the simplest amino acid. This is alanine and this is serine. In glycine, the R group is hydrogen. In alanine, the R group is methyl group, and in serine, it is CH2OH. Okay, this is given in the NCERT. You can see in your book. Then we have fatty acids. That is palmitic acid. So here the example of fatty acid, palmitic acid. We have discussed that the palmitic acid consists of 16 carbons. So you can see 1 carbon here, then 14 carbons here, and 1 carbon of the carboxyl group. So 1 plus 14 plus 1, 16 carbons. Then we have glycerol, that is trihydroxypropane. You can see 3 carbon molecules. All the 3 carbon are attached to 1 hydroxyl group. Then here we have triglycerides. Okay. 3 fatty acids are attached to the 3 locations of the glycerol. Then here we have the conjugated lipid, that is lecithin. Here we have the derived lipid, that is the cholesterol. So we have already seen the structures of the lecithin and cholesterol when we were discussing the lipids. So I hope you have understood this. Polysaccharides is completed. Now we'll start with the nucleic acids. Nucleic acid is the last type of biomolecules that we have to discuss. So nucleic acid is a polynucleotide. Polynucleotide. That means the building block of nucleic acids is nucleotide. So they are made up of nucleotide. Nucleotide. Okay. Now what is nucleotide? It consists of 3 parts. First one is a heterocyclic ring called nitrogenous base. Nitrogenous base. Okay. These are present as heterocyclic rings. Second part is a monosaccharide or sugar. And the third part is a phosphoric acid or a phosphate group. Okay, so these are 3 parts of a nucleotide: nitrogenous base, sugar, and then phosphate group. Okay, now these nitrogenous bases can be of different types. Okay, these are broadly classified into 2 types: purines and pyrimidines. Okay, so purines and pyrimidines, you can see the structures of them. There are 2 types of nitrogenous base which are known as purines: adenine and guanine. So adenine and guanine come under the category of purines, whereas cytosine, uracil and thymine, these are called pyrimidines. Okay. So as you can see that the purines are made up of 2 rings. 2 rings are there, whereas in pyrimidines only 1 ring is there. Okay. So adenine and guanine are called purines, whereas the cytosine, uracil and thymine are called pyrimidines. Okay. So we have seen that the nucleotide is made up of the nitrogenous base plus sugar plus phosphate. So one of these nitrogenous bases will be present. Okay. In one nucleotide. So here you can see this is the structure of the that is given in the NCERT. First one, you can see adenine, which is a purine, 2 ring structure, and this is adenosine. What is adenosine? When this adenine is attached with a ribose sugar, then it is known as adenosine. Okay, so when the nitrogenous base is attached with the pentose sugar, the sugar that is used here is the pentose sugar. That means consisting of 5 carbons, and that pentose sugar is ribose or deoxyribose. We have already seen the structure of ribose and deoxyribose both. Okay, so nitrogenous base, when they combine with the pentose sugar, they form a nucleoside. But this nucleoside is not the repeating unit in the nucleic acid. Nucleotide is the repeating unit. When this nucleoside, that means the combination of nitrogenous base and pentose sugar, is attached with a phosphate group, then it forms a nucleotide. Okay, then it forms the nucleotide, which is the building block of nucleic acid. Okay, so nitrogenous base plus pentose sugar will form nucleoside, and then nucleoside plus phosphate group will form nucleotide. So here you can see the first one is a nitrogenous base, that is adenine. It is attached with a pentose sugar. This is the pentose sugar, ribose sugar, right? So it is attached with ribose. Now it is a nucleoside and the name is adenosine. So whenever a nucleoside is formed, the suffix "ene" is at, "seen" is at. Okay, and when a phosphate group is attached to this adenosine, it forms adenylic acid, which is a nucleotide. Similarly, the other example is uracil, which is a pyrimidine. It is attached with this pentose sugar. It forms a nucleoside known as uridine. Okay, and then when the last figure is missing, similarly a pentose group will get added to this. It will be combined with the pentose, then it will be combined with the phosphate group, then it will form the nucleotide. Okay, so let me revise it if you have missed out something. The nucleic acid consists of nucleotides. Okay, the building block of the nucleic acids is nucleotide. Now what is nucleotide? Nucleotide is made up of nucleoside and phosphate group. Okay, nucleotide is made up of nucleoside and phosphate group, and nucleoside is made up of a pentose sugar and a nitrogenous base. Nitrogenous base and pentose sugar. Pentose sugar. The pentose sugar is generally ribose or deoxyribose. It can be either ribose or deoxyribose. We have seen the structure of ribose as well as deoxyribose in the polysaccharide. Okay, what is deoxyribose? When the oxygen from the second point is deleted in the ribose, then it forms deoxyribose. Okay, nitrogenous bases can be adenine, guanine, cytosine, uracil and thymine. Okay, now if the pentose sugar is ribose, then the nucleic acid will be known as ribonucleic acid. Ribonucleic acid. That is RNA. If the pentose sugar is deoxyribose, then the nucleic acid is known as deoxyribonucleic acid. Deoxyribonucleic acid. That is nothing but the DNA. Okay. Another thing that you have to note is the nitrogenous base in case of RNA can be uracil, not thymine, but in DNA it will be thymine. Okay. So other nitrogenous bases, adenine, guanine, cytosine, will be common in RNA and DNA both, but this uracil and thymine will not be common. In RNA only uracil can be present, thymine cannot be present, and in DNA only thymine can be present, uracil cannot be present. Okay, so I hope you have understood the chapter well. Okay, I have explained all the different types of biomolecules. First we discussed the protein, then we discussed the enzyme, which is again a type of protein. The exceptional enzyme was ribozyme, which is a nucleic acid. Okay. Then we discussed polysaccharides, lipids, and nucleic acids. Okay. So all the biomolecules are completed. I hope you have understood this well. If you have any doubt, please feel free to mention your doubts in the comment section. I will reply to your doubt, and if required, we can explain this again. Okay. So keep learning, keep scoring high. Okay. We will get back with another session. Thank you so much.